If you have the power to make someone smile, do it. This world needs more of that. A warm welcome and a fresh morning to everyone connected here to the first day of a international conference. Welcome and a fresh morning to everyone connected here to the first day of a international conference. A warm welcome and a fresh morning to everyone connected here to the first day of the International Conference on Biotechnological Effective Approach in Science and Technology, BEAS 2022, conducted by the Department of Biotechnology, K.S. Rangasamy College of Arts and Science, Autonomous, Tirchangood. Before we begin, I request all the participants to stay connected throughout the session.
A day without prayer is a day without blessing, and a life without prayer is a life without power. Now I request the Nightingales of Second Biotechnology to sing the prayer song.
நீராறும் கடலொடுத்த நிலமடந்த கெழிலொழுகும் சீராறும் வதனமென திகழ் பரத கண்டமிதில் தெக்கணமும் மதிச்சிறந்த திராவிடனல் திருநாடும் தக்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிளமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Thank you, girls, for your blissful song.
Sunshine is a welcome thing. It brings brightness. May I now invite Dr. G. Saravanan, Head and Assistant Professor, Department of Biochemistry, Ksrcas, to welcome the gathering. Sir, please.
சார் சரவணா சார் ஸ்டார்ட் பண்ணலாம் சார். சார் சரவணன் சார் சார் பேசலாம் சார். சார் சார் நீ ஸ்டார்ட் பண்ணலாம்.
நல்லா இருக்கா? Shall I start? Bold take it. Good morning, one and all present over here. It's my pleasure to welcome you all for this international conference on biotechnological and effective approach in science and technology, that is BEAST 2022. First of all, I welcome you. I welcome our beloved chairman, Dr. K.S. Rangaswamy, and Sri R. Srinivasan, Vice Chairman, and the Executive Directors, Mrs. Madam Kavita Srinivasan, in their absence here. And I welcome all the dignitaries, students, scholars, faculty members, and all the participants for this conference. And I welcome the invited speakers, Dr. Vidudalai or Regina, Dr. Kobalu Karnagaran, Dr. Satish Kumar, Dr. Sindhil Kumar Balakrishnan, and finally Dr. Rajiv Kangabam for this conference. Finally, I welcome you all for this conference in a grand success manner. Thank you, sir.
Thank you, sir, for such a warm welcoming words.
Life isn't about finding yourself. Life is about creating yourself. Now, I welcome a dynamic and vibrant person, Dr. V. Radhakrishnan, Principal of K.S. Rangasamy College of Arts and Science Autonomous, to deliver the presidential address. Sir, please.
அவன் நம்பர் மட்டும் கேட்டேன். ஏடிஜேபி அதான் கேட்டேன். 4838B. நான் இங்க மாத்தி எழுதிட்டேன். இது வச்சு அதை மிஸ் பண்ணிடாதீங்க. எல்லாம் ஒரு பக்கம் எடுத்து வச்சுங்களா கொடுங்க. மாறு சொல்லு.
Sir, yes, sir, you can start. Sir, you can start now, sir. யாரோ ஒருத்தர் ஃபோன் போய் கால் பண்ணீங்கன்னா வந்து ரெடியாக தான் இருக்கு. நான் சொல்கிறதே அவர் கேட்கல.
Good morning. I think I am audible. Bhupalan.
Sir, yes, sir. It's audible, sir. Audible, sir.
அதான் இப்போ எனக்கு ஒன்றுமே கேட்கல. ஏன் இப்படி? இப்போ நான் பேசுறது உங்களுக்கு கேட்குதான்னு தெரியலையே. என்ன சோதேட் எனக்கு தான் பாருதா? ஐ பின் கனெக்டட். எங்க பிரச்சனை வருது? அப்போ நான் பேசினா கேட்குமா இல்லையான்னு எனக்கு தெரியலையேப்பா. பாருங்க, போட்டு பாருங்க, நீங்க கனெக்ட் பண்ணி பாருங்க ஒரு நிமிஷம். ஹலோ, am I audible? ஸோ வீடியோ வருது, இதுவும் வருது, ஆடியோ வர மாட்டேங்குது. ஆடியோ மைக்கு கனெக்ட் ஆகுது, மைக்கும் கேமரா. am I audible now? Okay, 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 okay. Good morning, friends. Respected chief guest of today, Professor, the invited speakers, respected invited speakers, Dr. Vidudalai Regina, Dr. Gopala Karunagaran, Dr. Satish Kumar, Dr. Sendil Kumar Balakrishnan, Dr. Rajiv Kangabam, and head of the Department of Biotechnology, organizers of this event, and my dear participants. We know very well digitization is one big boom, and it has got its own setbacks also, and we, at the same time, we enjoy both the things, the benefits and the setbacks. That's what I would like to say on this day. The title, Biotechnological Effective Approach in Science and Technology, is what you can say, relevant to this situation, because we have seen the boon and ban of science, especially the field of biotechnology, and we have been affected, and we have, I should not use the word enjoyed, but we have been affected by the effects of this double-edged sword for the past 2 years. The pandemic is one best example for that, one big example for that. And I am sure programs of this level and programs of this volume should be organized regularly to make people understand how we can use effectively the biotechnological concepts in the field of science and technology for the betterment of man, rather than for the destruction of mankind, not only mankind but also the environment. And I am sure that the key takeaways that we are going to receive from the respectable speakers will be useful for us, and we can, I mean, as a part of this event, I would like to say we, and we can use these concepts in our field to make ourselves better and also to make our world a better place to live in. I wish all the best for the program, and also I welcome all the participants, delegates, the invited speakers, and everyone on behalf of the KSR College of Arts and Science Autonomous, and also on behalf of the KSR Education Institution. Thank you for giving me this opportunity. Thank you and good day.
Thank you, sir, for your valuable speech. Never stop learning, because life never stops teaching. Now I request Dr. S. Ramarajan, Organizing Secretary, BIS 2022, Assistant Professor, Department of Biotechnology, Ksrcas, to give an overview about the conference. Sir, please.
A very pleasant good morning to everyone. As we are about to kickstart the event, it's my pleasure to walk you through the conference, introduce the speakers and delegates from various countries, and to share their knowledge and a widen mental horizon. Today conference has about 540 participants from more than 37 colleges and universities across India and abroad. which was possible only through the advancement of internet and virtual meeting platform of in spite of pandemic and situation. Nowadays, conference and connecting with scientists across the globe and accessible in single click. We are proud to act as a platform to connect you with our now handpicked panel of speakers. As we live in the biotech era, the conference titled Biotechnological Effective Approach in Science and Technology BIS 2022 aim to bring together scientists, students, and research professionals to exchange the scientific ideas. It would be also serve as a platform to budding scientists and undergraduate students to explore their biotechnology to by biotechnology from various countries like Singapore, China, South Korea, etc. The major outcomes of this conference we expect from the conference is ample exchange of knowledge by enlightening participate on research trends, various fields of biotechnology. We request fullest cooperation of the participants to make this 2 days conference of a fruitful one. Thank you.
Thank you, sir, for a wonderful and thoughtful speech.
No more duties. No duties more urgent than returning thanks. Now I request Dr. S. Bhupalan, Organizing Secretary, BIS 2022, Assistant Professor, Department of Biotechnology, Ksrcas, to propose the vote of thanks. Sir, please.
Thank you. A very good morning to everyone. It is my privilege to propose the vote of thanks at this occasion. I, on behalf of the Department of Biotechnology, K.S. Rangasamy College of Arts and Science, the entire crew of Beast 22, and on my own behalf, I extend a heartful gratitude to all. I from the bottom of my heart thank the college management lion Dr. K.S. Rangasamy Ayya, founder chairman of KSR Educational Institution, and through R. Srinivasan, vice chairman, for their constant support in all aspects. Thank you, sir. I register my profound thank to our dynamic principal, Dr. V. Radhakrishnan, sir, for his motivation to take up such academic activities. Thank you, sir. I extend my deep sense of appreciation to Dr. M. Prasanna Rajesh Kumar, sir, Head Department of Biotechnology, and also I would like to appreciate Dr. G. Saravanan, Biochemistry, Ksrcas, for his valuable guidance and freedom to perform all our academic duties. Thank you, sir. I render my special thanks to all the faculty members of biotechnology, microbiology, and biochemistry family for playing key roles in organizing this event. I am thankful to our honorable speaker who have consented to deliver talks at this conference amongst their hectic schedule. I record my sincere thanks to the head and staff members of ISCOM department for providing their technical support. I place my sincere appreciation to the research scholar, PG students and UG students of biotech department for playing active roles. And a huge thanks to all the participants, including faculty members, research scholars, PG and UG students from various colleges and universities from India and abroad, who are a big part of this conference. A big thanks to all of you. Thank you, one and all. Last but not least, I would like to conclude with the most popular Tirukural quote. அறிவாற்றாம் காக்கும் கருவி செருவார்க்கும் உள்ளிழுக்கல் ஆகாரன் பொருள் அறிவு என்பது அழிவு வராமல் காக்கும் கருவியாகும். மேலும் பகைவராலும் அழிக்க முடியாத உள் அரணும் கோட்டையாகும். எந்த சூழ்நிலையிலும் அரண் போல், அதாவது கோட்டை போல் நின்று நம்மை காக்கும் என்பது அறிவு. எனவே அறிவாற்றலை வளர்த்து கொண்டால் நாம் வாழ்வு வளமாகும், நம் வாழும் நாடும் வளமாகும். ஜெய்ஹிந்த். தேங்க்யூ.
Thank you for expressing your heartfelt gratitude, sir.
Great achievement requires personal force, determined spirit, and self-confidence. I'm glad to introduce one such young and eminent scientist, Dr. Vidudalai R. Regina, Senior Research Fellow, Biofilm Biology Cluster, Singapore Center for Environment, Life Science and Engineering, Singapore.
Dr. Vidudalai has earned his PhD in nanoscience from RS University, Denmark. He has been a postdoctoral fellow from 2013 to 15 at Interdisciplinary Nanoscience Center, iNano, RS University, RS Denmark. His area of research includes human skin microbiome and host microbiota interactions. Sir is a microbial ecologist studying human skin microbiome in pursuit of understanding the eco-evolutionary dynamics of living things in communities. Working at the interference of industry and academia with a force on translational research and product development. He was a founder of biotech company to offer consultation service on microbial diagnostic, quantification, and quality control assay development for food industries and terminated this activity since he moved to Singapore. He has been a JRF in NSS Pune and National Institute of Ocean Technology, Chennai. He has worked on various research projects and has attracted research grants from various sources. He has received $250,000 from Ministry of Education, Government of Singapore under the call Academic Research Fund Tier 1 and many more. He has delivered many lectures in different scientific forums and scientific meetings. To his credit, he has obtained 2 patents and he published a number of research articles in international peer-reviewed journals. From his schooling and college days, he is a recipient of various awards in both academic and extracurricular activities and has actively volunteered in NSS, etc. He is a socially active blogger, voracious reader, and science enthusiast with interest in the sky gazing and martial arts. We are delighted to have such a multifaceted person to deliver a talk. With this brief intro, now I invite Dr. Vidudalai R. Regina to enlighten us on the topic Meeting the Microbes with Technology from Investigation to Innovation. Sir, please.
Okay, thank you for the introduction. You guys hear me all right? Can you hear me? Can somebody say yes? Okay, all right. I'll share my screen first. I hope you can see my screen now. Okay, great. Okay, cool. All right, thank you for that introduction. And give me a second. I'm just going to pull my stuff here so okay. So, of course, you guys introduced me. I have a slide for myself, so I'll just go through it quickly. So I did my bachelor's in zoology in ABC College, Mailaduguri. That's in old Nagapattinam district, which is Mailaduguri district at the moment right now. And I did my master's in St. Joseph's College, Trichy, in biotechnology. And then after that, I wanted to do research, but then for a short time, I was like writing exams and so on. So I was also working as at the Tata Age Insurance for some time. Then I joined National Institute of Ocean Technology, where I basically started working on microbial biofilms, bacteria, etc. And then I moved to National Center for Cell Sciences as a junior research fellow under the DBT ministry. Then, because I was doing a lot of work in India for some time, I wanted to do a PhD abroad. So then I went to iNano, which is Interdisciplinary Nanoscience Center in Aarhus University, where I did my PhD in basically in nanoscience coupled with microbiology. So that was a very good experience, cross-disciplinary study. And then after PhD, I went to Delft in Netherlands. I was living there for a couple of years. But mainly, I wasn't working or anything. I was writing some papers, but then mostly, I took a career break, because my wife was pregnant at the time. She delivered our first baby, and she got a job also with pregnancy package in the Netherlands. So I thought it was the rightful thing for me to take a break and take care of the baby. So yeah, so that was a very good time, very good break, as well as had a good time enjoying the family. And then I moved to Singapore in 2016 as a research fellow, and then I got promoted as a senior research fellow. Now I'm also a deputy director of a new research collaboration between CELSI, which is the institute I'm in, which is in NTU, Nanyang Technical University, and L'Oreal, which is a cosmetic company most of you know. So they are into skin research and the skin microbes and all that. So I'm directing research in that center at the moment. So so today's topic for me is like meeting the microbes with technology from investigation to innovation. So this is a mouthful, of course, and what I want to do is to give you guys a little bit about what is discovery, which is basically investigation, and then what is innovation, what are the differences, how things happen from discovery to innovation, etc. briefly. And then I'll touch upon two examples. One is with genome editing, which is basically out there, everyone knows. So I'll give the example for the discovery to innovation story using the genome editing theme. And then I will talk about skin microbiome, which is basically my expertise and what we are doing. And I should tell a disclaimer here that because some of the work that we are doing is under confidentiality, I won't be able to discuss too much details into it. But I'll tell you how we are approaching the work and what we are discovering and how we are translating that discovery into an innovation and also products, etc. Right? And then finally, we'll see a little bit of a perspective. All right. So so you guys are mostly from the academic institution, right? It's a college. You're studying bachelor's. Most of them are bachelor's, masters, and PhD students. And by the end of your masters or even your PhDs, many people, including myself, had this problem, like where to go. Should we continue in the industry, or should we stay in academia, teach, or do research? Research, if it is research, where you want to do the research? Is it an academic institution like universities or public research institutions, or you want to go into an industry, a company, do research for the company, and develop products, etc. So one thing I want you guys to understand is that there is nothing like one is greater than the other, or one is lower than the other, right? Of course, when you are in the university setting, you are paid pretty less compared to industry setting. But the thing about this is these are two different models, right? So in university is basically a model where the government invest in the human resources and developing the intellectual property of the whole country, whole nation, the next generation, right? So it is an investment model. So you put in money, you collect tax, and you put in money into the institutions and develop, grow the students, make them good enough to be able to do discoveries and innovations that will actually take the country forward, to take the humanity forward, right? So those people, either they stay in the university and do that investment portfolio further by teaching, by doing research in the institution, etc., or they move to the industry and actually do innovations, make really nice products that people can use, and that is actually impacting the society, and the revenue collected from this is actually coming back to the government as tax, which is actually invested back into the university, right? So both these institutions are really important. And for me, I don't want to go to the industry because I feel like discovery is the thing that actually excites me more, because both the institutions do discovery as well as innovation. But in the academia setting, in the university, discoveries happen a lot because we think about basic sciences, fundamentals, all that. We do innovation based on this discovery, right? If you discover something, then we do innovation. But in the industry context, discovery is like very less. They don't really care about discovery because discovery is not going to make profit because that institution is profit-based institution, right? So somebody start a company because they want to make a profit out of it. So the idea here is that they want to innovate. So what they would do is they would actually go for whatever things have discovered before. They will look into it, they'll study it, and then they will actually populate ideas to find out how that knowledge can be taken into innovation, right? So those are the two different models. I'm spending a few minutes here because many a times when people finish their studies, they don't know what to do, right? And most of the time, what we are propelled to do is that they think like, okay, where is the money? Where you will get more paid, etc. Right? And then at some point, people will complain about their job. I don't like this job. My manager is this, that, and all that. Right? So you guys are young, so try to think about what excites you more. Are you excited about making something new, or are you excited about learning something new that is never discovered before, right? So there are pros and cons on both sides, but the most important thing is what actually excites you more, because you're going to do this thing for your life, right? The entire life. So it's a good idea to make sure what really makes you happy. Okay, so leaving it up there, so the investigation or the discovery part generally takes a really long time, right? And the innovation part basically takes shorter than the discovery part. Innovation also can take long. For example, I'll give you an example of Dyson, right? So Dyson is a company, it's a very new company, and that has actually grown really fast. If you remember about these vacuum cleaners, right? The old type of vacuum cleaners are like, they don't really perform well and so on. And then Dyson came up with this cyclone technology where they were able to, maximize the suction amount, etc. And based on that, they have made tremendous progress, and now it's a global company, right? Just with one product. So, but for that one product, the first prototype, it took like 5,500 plus prototypes to make that one final Dyson product, which actually made them to be, to reach this, heights. So it does take time, but when compared to discoveries takes even longer, because discovery comes from curiosity, and you dig something, you find something, you don't find something, so it's a long process. So sometimes for something to come around to innovation stage, it might take 10 to 20 years for a discovery to shape up and have enough knowledge about it, and then you can actually use it as an innovation. But there are many things that are already out there, and you can use it, and then you can actually innovate. So that's an easier thing, right? So what I'm going to do is I'm going to give the first example as the genome editing, and I'll specifically talk about the CRISPR technology, which is a very recent one, and many people know it won a Nobel Prize, etc. So we will talk about I'll talk about the CRISPR technology and how when it was discovered and how it evolved, and then now we are in innovation stage at which we are applying for genome editing, etc. Right? So just to give an overview about the genome editing applications, right? So you can apply the genome editing in many different things, right? So you can do gene surgery, you can do drug development where you can engineer the microbes genome to synthesize specific biochemicals that are used as drugs, or you can make animal models where the drugs can be tested. Also, you can make genetic variants and materials that can be used. There are many research that is going on to replace plastic like non-degradable materials rather than making biodegradable materials from biological organisms. Right? So those things, and of course, food, GM crops, fuel. So these algal-based fuels where the algae and these microorganisms can be engineered, so then they can produce a lot of ethanol and methanol and these kind of fuels, which can actually which can be used as a sustainable source of fuel rather than petroleum products, which will eventually dry up, as well as it is actually causing a lot of global warming and all that conditions, right? So thing is, with the genome editing, a little bit of a basics, like what happens in the genome editing. Generally, genome editing is basically done where we cannot go specifically into editing things, right? what we can do is we can actually either split the DNA, and then so we have repair mechanisms in our cells, right? So if the DNA is broken in any place and the genome is broken, and then it will repair itself, right? So it will actually patch up things. So there are two types of repair mechanisms that actually helps in this process. If a double stranded break happens in our DNA, the non-homologous end joining, what happens is it basically, if there is a break in the DNA, it will just try to join the pairs, join the ends. So that's the non-homologous end joining, right? And then homologous directed repair mechanism. This is a little bit different. Where, okay, I'll give you an example from with a book example. Like if you go to a library and you see that there is a you take a book and you see that there is a page missing, right? What you can either do is you can just make sure that you remove the page completely and try to join it, or you can actually go to the same library, find another book that is of the same kind, and find out what is the passage that is missing, and then copy that passage, either write or print, and then put it into the in the page where it is supposed to be, right? So that's what the HDR mechanism looks like. What happens is this part. So the mechanism actually looks for reference material basically, and then it will actually repair precisely based on the reference nucleic acid fragment. So those are the two mechanisms that actually happens. So how we use this for genome editing? We basically come up with enzymes that can actually do this double-stranded break and let the cell take over the repair mechanism. And this is actually a little bit of little bit detail about how it is done. I'll just go through it. If you have doubts or something, you can ask me later. We can discuss about this. So for example, there is a break in here, and you see that this particular area CTT is gone, TG80 is gone here. So what happens is there is a ligase that actually attaches the two fragments, and then there are polymerases and nucleases. They will just come in, and then they will read what is A here, and then it will add the T here. If it is a T, it will add A, C, G, A, T. Similarly, it will just make it. And what happens in this case is, because it cannot actually, it doesn't have a reference. It is an error-prone method. So the resultant can be like this, or like this, or like this, right? So it depends on what kind of shear it has done for the DNA. So there are multiple things that are happening. People have researched on that, how this is happening, etc. And then homologous end repair is basically like this. So it basically, if there's a break, and then you have a template, which is a donor template, and then either it is inserted, or it will just use it as a reference and fill up that particular region. So then it is a very precise editing, right? So this is how it works. Now, so as I said, basically for genome editing, only thing that we do is cut the DNA. That is our only aim, because the rest of the thing is taken care by these cells, right? So you can see this here, there are different types of nucleases that have been used in genome editing. Meganucleases are the nucleases that previously people were using a lot. These are really large nucleases that have been isolated from microorganisms and other things, and they will just put it into the cell, and then it will just go and cut, right? But these ones are really not very precise. It will cut anywhere, and we cannot have much control about it. So what will happen is we are going to generate a lot of different mutants, and then we have to screen for them and identify the ones that we are interested in. And then the zinc fingers came in, and what they did was they found that these zinc finger nucleases, they have this FOC1, which is actually the nuclease that cuts the DNA. And these are basically subunits of proteins that has a 3 nucleotide recognition sequence, right? So based on this, what you can do is you can actually engineer a protein with these subunits which have specific binding sites. So let's say you want to you want a region that has ATG, GAT, TTA, GAT, whatever it is. Then you can actually basically engineer it in a way that they are randomly repeating and not repeating, randomly placed next to each other. Then they will actually go recognize that particular sequence and then bind it, and then the fork one will actually cut it, right? So that's how it works. However, this is also based on 3 nucleotides, right? So you can actually do only there is also limitation that it cannot be very precise. You have to go with what is available as a subunit, right? Then the talent came in. Talent basically actually these are subunits that have single nucleotide recognition. So this is more easy now, right? So if you need TAAGT, GGT, then you can basically engineer these subunits in a way that they are complement to that particular sequence. Now fork one is also there. Now it is a little more precise. Then it can actually find the right region. It will bind there, and the fork one can cut, right? Now, so these are all protein based, and you can see that this is a feasibility kind of thing where the talents are the most useful, because this is more precise compared to the zinc fingers as well as the meganuclease. But these are proteins, right? So these are proteins, so which means it's a little bit protein DNA interaction can be a little bit hazy, but CRISPR came in where it is nucleotide interaction, because you have an RNA that actually goes and find the DNA. Because these both are nucleotides, this is more precise. So that's how this came in. I'll explain how this is working, but this is how the innovation actually went on for gene editing, right? Okay, CRISPR. Let's first see the investigation part, right? So what happened was, of course, Emmanuel and Jennifer, they got the Nobel Prize for the CRISPR technology, and this is actually the paper they published in 2012 in Science that actually, got the attention, and further, the Nobel Committee, identified them as the inventors and gave the Nobel Prize. So this broke a couple of ceilings. Basically, I just want to emphasize that one, this is the first Nobel Prize where a pair of female scientists are getting a Nobel Prize. So this is huge in the human history itself, right? So that's one. The second thing is, this is the fastest. I think it was like less than 8 years or something. It was the fastest Nobel awarded. Usually, Nobel takes a long time to get awarded because the application should come out, and then if it is so much useful, then only people will get awarded. So that way it is very very good. So just to give a perspective, the CRISPR thing is not a new one, right? It was actually discovered, the presence of CRISPR-like elements in the genomes of bacteria, especially E. coli, was discovered in 87, and I assume most of the students here were even not born that time, right? And then you can see that in 2012 was the one that they proposed the characterization of the proteins, etc., and the application actually followed, right? So you can see that it took a really long time, like 87 to 2012. It's roughly 20 plus years, right? So that's why I said like it takes decades, 10 to 20 years to form something before innovation can happen for really new stuff. So this is just to highlight where this innovation comes. Okay, so the investigation part, this is what people were interested in. They were curious about like what is happening in the CRISPR thing. So in nature, what happens is the bacteria, the CRISPR system is actually the immune system for bacteria. So in our system, what happens is we have our immune system, and let's say, okay, that's why we use vaccines, right? So they are actually using them as vaccines. Bacteria are using CRISPR system as vaccines. So what happens here? So in our system, if a virus comes in, what does our immune system do? It recognizes the virus. One, it kills it, neutralizes, and get it away. The second thing it does is actually it takes part of the vaccines, part of the viruses, or whatever pathogen, its protein structure or whatever, and then it actually makes a memory out of it with B cells, etc. And then when the thing comes next time, it will automatically neutralize it easier without getting sick, right? So that's the basis for vaccines. So this is actually happening in our own cells naturally. So what we did was we actually use attenuated viruses or virus proteins and DNA, and then we put it inside the cell, so they recognize it as a potential threat, and then they produce a lot of antibodies, and then it is there for lifetime. And in many cases, it is not there for lifetime. You know about COVID, the thing is going on now, also common flu. So there are coronavirus families which are kind of very stealth like, which yeah, so that's another discussion. But then this is how the immune system in human works, right? So similarly, bacteria has this CRISPR-Cas system. What happens is when there is a virus that is attacking, the viral DNA comes in, and then this CRISPR genes, they go bind to the viral DNA, they cut them, they recognize a specific sequence, and then put it into this CRISPR array. These are called spaces. So all these different colors are actually previous viral infections. So when the new virus comes in, it will actually recognize this region based on the spaces, and then it will basically cut those regions using the Cas9 protein. So basically, it is actually, it takes it. Okay, I think I can give you a better thing here. So this is how it works, right? So it recognizes the virus, and then it cuts the DNA so that it will not get infected. Now, there are multiple types of CRISPR-Cas system that is available, and one that was very useful was the type 2 system, which has this Cas9 gene, which is very, very specific, right? So let's come to the innovation part. So these are investigations. So we know how CRISPR work because it has this DNA where these regions are already captured, and that region is already ready. And when the virus or whatever comes in, if the region is a match, then the protein goes and cleaves it, right? Now, how can we use this? We can basically program it in a way that we can program the spaces, right? So these are the CRISPR RNA is the spacer that we can program, and this tracer RNA is something that is attached to the spacer that will actually hold the spacer region, and also this is the region where the Cas9 will bind, right? So when these two tracer RNA and the CRISPR RNA are together, it's called guide RNAs. Okay. Yeah. So basically they have made a linker, so it is attached to each other. So this is called the guide RNA. So what happens is, now let's say this is your target region, the blue region, right? And this is the region that you want to cut. Usually the PAM is actually the protospacer region motif. And this is actually the region which is recognized by the Cas9, and it will cut after 3 nucleotides exactly, right? So what you will do is you want to design this guide RNA where the tracer RNA is combined with the CRISPR RNA. This blue region is what you are designing. If you want to cut this region instead of this, you're going to design something that is actually complementary to this region, right? So then it will change. Then the whole Cas9 will actually bind here, and then it will cut here, right? So this is what actually the engineering or the technology actually transformed to. So only thing that what we did was, this CRISPR RNA, naturally, it is acquired from these viral genomes and then placed inside the cells. What we are doing is, we are actually creating this region depending on the region that we want to target inside the genome. And then we put this inside the cell, and that's it. It will do the job. So how does it actually practically work, right? So let's say you're designing an experiment, you know which region to cut, etc. So what you basically do is, you will design it in a computer, like, which is the region, etc. And then you synthesize that molecule, and you basically put it into a plasmid. So you have this guide RNA, and then you have the region that is responsible for synthesizing the Cas9 protein, and it is placed inside a plasmid and then thrown into the cell. When it is inside the cell, what happens is, this thing goes in, and then it will basically cut the region where you want to make a cut. If it is a mutation that you want to do it in the DNA or whatever, it will cut. Or if you want to add something, insert something, you're going to also supply a guide RNA. template RNA, right? So that is what it is going to use, and then it will synthesize that region so it can insert it, right? So after that, what you will do is you're going to generate a lot of mutants, and then you'll basically do a PCR or something like that to see which are where exactly these mutations have take place, and then you can isolate that particular cell and then expand them and then use it for your purposes, right? So this is how it works. I hope you understand. If you don't, do ask me at the end. I'm happy to explain it again. So as I said again, I just want to put this into perspective one more time that, it took more than 20 years from the discovery of the system in one bacteria. And then if you see, these are, they found it in multiple bacteria and then other families, and they found that it is widespread in prokaryotes and all that. And then they find out the CRISPR-3, all these type systems. And then finally, in this century, that they were able to, come up to a level where they can say, okay, if we modify this system like this, we can actually use it to use it for our advantage, right? Okay, this is just one thing that was proposed for gene editing, right? So it is just cutting or placing something inside. So that's the only thing that people were doing. However, innovation kicks in here. Now you have the knowledge of how things work, right? So what people did was they took Cas9 and as well as Cas13. Cas13 is another kind of protein, similar thing. It will do. Only thing is it will actually recognize the RNA, and this would recognize the DNA, right? So that's the thing. So what they did was they used so this is what we just saw, gene editing, right? So you can particularly specifically edit a gene, and then there is another thing. What they did was, it's called dCas9. dCas9 is actually dead Cas9. What does it mean is, the Cas9 has two functions. One, it will bind to the DNA specifically to a region where the spacer has asked it to bind. Two, it will cut specifically, right? Now dCas9, they removed the cutting part, right? So it is dead in a way that it will not kill. It will not cut, but it will bind to the DNA. Now there is a lot of things that people can do. Say for example, so gene regulation. What happens if there is let's say if there is an operon or whatever, if there is a gene that you need to induce, and then sometimes the induction requires a particular effector or a protein or a molecule, right? So you can basically attach that effector to the Cas9, and then you can program it. Basically, you can design the spaces so that it can bind exactly next to the gene that you want to regulate. And if they bind, because the effector is close to the gene that you want to regulate, it will start expressing or suppressing, right? So that's one. And then another thing is epigenome editing, where epigenetics is basically there are modifications on the DNA bases, methylation, etc. And what you can basically do is you can actually tweak that, because methylation is something that is very chromatin imaging, right? So let's say you want to image a particular gene of interest and understand where that gene is in the chromosome. We have multiple chromosomes, right? Depending on which species we are. Like, of course, we are humans. We have 23 pairs of chromosomes, and other species have other number of chromosomes. And if you want to identify which chromosome something is, then what you can basically do is you design the particular gene sequence into the dCas9 system, and then attach the dCas9 protein with a fluorescent marker, and then it just basically binds there, and then you can do chromatin imaging. Let's say this gene is actually present in this chromosome, the red one is present in this chromosome, right? Now, you can also do chromatin topology, right? So topology is something like, the DNA is pretty long, right, compared to the cell's size. But then they are actually, crumbled together, and they are in a really compressed form, and that can also affect how the gene regulations are or the expressions of certain parts of the genome can be controlled. So you can also do that by using these kind of Cas9s, where you can actually bind them. You can actually attach these protein dimers. One half of the protein, I mean, the complementary, one complement is in one Cas9, the other one is in the other one, and then these both can bind to this particular region. And because they are complementary to each other, they're going to pull this and then attach. So you're creating a loop, right? So these kind of modifications also you can do. And Cas13 is actually similar to Cas9, but then this is RNA targeting. So this is specifically targeting the RNA, not the DNA. Let's say you want to control a gene expression at the transcriptome level, but not in the genome level, then you can also do that. It's a little bit easier to do that as well, because you don't have to mess up the genome. You only want to, let's say if there is a disease condition where a particular gene is continuously expressed and you just want to suppress that particular expression, then you can actually use this kind of method. The next thing is actually, I'm sorry. Yeah, so the next thing is actually the base editing, which is, again, it is a similar situation. You engineer the Cas9 to go to a particular place, and then you have certain, functional, I mean, you have certain things that, let's say you want to take out a T and then put a G in it. It's a single nucleotide modification, and you can basically attach that particular modifier to your Cas9, and then you throw it into the cells, and then that will do the job, right? So you see that there is one thing that was discovered, and then there is a plethora of applications that you can actually get into, right? So this is the power of innovation. Okay, so now I think I have convinced you that how things work, and in a way to, you hopefully you understood that how discovery works and then innovation works. Innovation faster than the discovery, and how things work like that, right? So this is a previous example, and I'm going to give you one example from my work in our lab, where the skin microbiome. So we are interested in skin microbiome, and we want to investigate the skin microbiome to understand how it is actually helping us, and is it actually associated with disease conditions, etc. Right. So what we wanted to do was that to basically sample a lot of human beings in Singapore. Basically, we did this study, and then we wanted to understand, how the healthy people's skin microbiome looks like. But okay, before going details, microbiome is basically a collection of bacteria, fungi, all other microorganisms on our skin and inside our skin, right? So we are all populated with a lot of bacteria, and this is a relatively new knowledge. And then what we know now is that there are many disease conditions where the disease condition is associated with this change in the skin microbiome. So let's say if this is a healthy microbiome, where a healthy person has a lot of different types of microbes, and let's say if he encounters an infection, right? This is a new microbe that is coming into the system, and then it actually changes the state, which is called a dysbiotic state, which is also a disease state, right? So what happens is that this is an example of a healthy skin, where you have the skin part here, and then the skin microbiome, which is a lot of different types of bacteria and other things. You see that the red ones are staphylococci, and the green ones are firmicutes, etc. But in the disease condition, what we see is, let's say, for example, atopic dermatitis or eczema, where people have rashes in their in their hands or whatever places, mostly the moist areas. You see that the staphylococci bacteria goes up, which is very red, and then it is very itchy, flaky, and all that. And similarly, we can say that this is a healthy hair follicle, and hair follicle is everywhere on our skin. And the hair follicle is also colonized by a lot of different bacteria. And if the composition changes, we also see that acne, which is a pimple we call, right? So this is also associated with the bacteria called Cutibacterium acnes. The old name is Propionibacterium acnes, and they causes this pimple. So we are trying to understand actually what is happening, why this shift in the microbiome happens, and so on, right? So we know that our body is colonized with a lot of different microbes, and you can see these color codes are different types of microbes, and different areas of the skin has different types of microbes. The area or the region is basically defined by how sebaceous or oily the region is. Let's say, for example, or face or scalp, these are very oily, or we call it as sebaceous, or moist, where you have this armpits and all that areas are moist, and then you have dry, right? Palms are dry, your legs are dry, and so on. And what we found was that these are the only three organisms that are majority of the microbiome consist of, which is Propionibacter magnes, Staphylococcus, and Malassezia, which is a fungi, right? So these actually constitute about 90 to 95% of the whole thing. So we understood that if it is a disease condition or the healthy condition, these are the bacteria or the microbes basically they are responsible for how we work with. So what we basically did was we created a laboratory model where we identified these three organisms, and then we basically built a system to culture all of them together. And then also we used fluorescent in situ hybridization like methods to identify them when they are together in a system. Now, the problem we encountered a lot of problem. It took 2-3 years to come up with this media, etc., because all these conditions are different. So this bacteria is it does not grow under aerobic conditions. This bacteria grows in both conditions. This bacteria also need a lot of lipids, etc. So finally, we came up with a medium, and then what we did was we tried to understand how they are interacting in the system. Because all these bacteria are present in one place, so we know that everything is secreting something, everything is eating something, and they have a lot of interactions going on. So without going too much into details, we kind of found out what is actually produced by which bacteria, and if this is consumed by the other bacteria, and if it is consumed by the other bacteria, is it benefited by it or is it prevented by it, right? So when we worked out this, and we have a full map of this thing now, which I cannot really disclose it at the moment, but what we found was that depending on the skin condition, healthy or unhealthy, we see that one of these microorganisms take hold and they secrete something that will enrich a particular bacteria, also the skin, our skin. Also, they will actually prevent certain types of bad microbes from growing. And also they will actually induce antimicrobial systems of our own skin, right? So they are actually they are basically working in collaboration with our immune system to prevent infections and so on. So once we mapped out all that, the applications are like a lot, right? So the next thing what we are doing right now is to build a host microbiota model, which is basically our own skin and bacteria, and to understand like how these interactions are going on. And because we can actually understand this interaction, we can actually go for new drug discoveries for specific conditions, as well as we can actually use this metabolites that specific bacteria and the other microbes are producing. We can do diagnostics and see like which organism is more in particular area and so on, right? And also let's say there are raw materials that pharmaceutical industries and cosmetic industries they are interested in, which they want to find out if these raw materials are actually acting on these microbes, and if it is acting, is it beneficial or not. So those kind of diagnostic systems that we are developing. And also we can have probiotic and prebiotic formulations that we can develop. Probiotic is basically live microbes that you can actually apply as a as a cream or as a system. And prebiotic is basically you supplement some chemicals with which the bacteria or the microbes can actually grow well or prevent them from growing, right? Also, we can do a little more discovery into microbial ecology to understand how this dynamics works in an evolutionary scale, why this dynamics actually happening in our skin, how we and these microbes are related in an evolutionary sense, and all that, right? So I'm going to give you a couple of examples here very quickly. I know the time is running out. So a prebiotic that we are working on, it's going to be in the shelves very soon. And there is one a probiotic which is recently developed and launched in Belgium, where one of our previous scientists in our group actually is part of the discovery innovation, right? Okay, so innovation part. So this is a problem. So we have a condition called seborrheic dermatitis, where it is actually creating itchy skin and all that. This is basically because and also rashes in other places of the body. These are basically because of these two microorganisms, which is the fungi and Staphylococcus. These two actually grow really a lot than the amount there that should be in the skin, right? So this imbalance in the microbiome causes this condition basically. So what we found was that because we have now worked out these network mapping, what is doing what, we identify specific molecules that can actually prevent some of these microbes from growing, right? So if you put these microbes, let's say these are the microbes that are growing, and if you put this particular chemical or the molecule, then these microbes will not grow, right? So based on that, what we did was, how do a product development look like? So we first patented it, that this particular molecule can be applied, and this is a molecule that can be synthesized, but it is a naturally occurring from these microbes. And then right now what we are doing is we are actually developing formulations, and we are testing it on people. And if this thing goes very well, and then we will soon be able to launch it into the market, right? Which I hope, like, probably by the end of the year or something, it will happen, and yeah, people can use this product when they have this particular conditions, right? Another example, which is already on the shelf, which is very exciting, which is so this one is prebiotic, right? So this is a molecule that will actually, it's a chemical molecule which is naturally produced by the microbes on our skin, in a healthy skin. So what we are doing is we are taking that chemical and putting in unhealthy skin, so that skin becomes healthy, right? Because they are affecting the bad microbes. Now, another example is actually, right now it is on the shelf. This was launched by Biomedical in Belgium. It's called Live Skin Probiotic, which is basically one of the very good bacteria, very beneficial bacteria on our skin. So they basically made a formulation that you can apply on your skin. So think about it. You're applying basically live bacteria on your skin, and the clinical trials have given tremendous results and so on. So this is another example for the innovation, right? So this is what basically I want to emphasize again, from a perspective side, that it takes time for discovery, but then if you investigate something, then you understand the system very well, that you can innovate and make a lot of different things. Now, from a perspective side, I said all good things about CRISPR and so on, right? But also you see all these news out there that there are also a lot of concerns about CRISPR, because it's also error prone. And if you see, this is a clinical trial directory. If you you can go check this CRISPR news medicine, and also CRISPR based clinical trials. There are hundreds of trials going on at the moment, right? But again, even though they are trying to come into the market and on all that into the clinics, there is also a lot of concerns about it, and it should be regulated, right? So I said these are the two things that I said, right? Discovery and innovation. But you guys also should think about going to the next step, which is regulation. This is also very important, right? If you don't regulate certain things, it will really fire back, and we don't have so much regulations at the moment. I mean, there are regulations, but then for new discoveries, for new products, new innovations, more regulations are coming in. So students who are doing science, you can choose discovery path, you can choose innovation path, or you can also choose regulation path, which is not very popular at the moment, because not many people know about this, right? So you guys should learn about this, find out what you need to study to go into these paths, and I wish you all good luck. Just before closing, I just want to share one thing, where we are actually a team of people from different countries like Germany, Singapore, UK, etc., also India, that we are hosting something called Sevi Kunavu in Tamil. It's basically, we are inviting scientists from around the world who are basically from Tamil Nadu, and we are running this in Tamil because it's easy to communicate science in Tamil. And we are trying to do this for the student community, so you can actually interact with these scientists actually, or you can know like what are the paths that you have for your future, etc. So you can visit us at facebook.com seviunavu, seviunavu, or you can visit my webpage drvidu.com. You can get connected, and we can let you know whenever we have these sessions going on, and you're very welcome to join us. Right. All right, with that, I will close this. Thank you. If you have any questions, I can take it. Thanks a lot.
That was really an amazing session. Thank you, sir, for sharing your great knowledge with us in a very clear and interesting way.
Thank you. Thanks a lot.
The session is open for discussion. Participant can post your queries in the comment box.
I encourage everyone to don't be shy. Just ask any question. You don't have to ask a question about the subject we talked about. You can ask anything about your studies or what to do next, future, whatever it is. Right? It's a good opportunity for you guys. நீங்க தமிழ்ல எழுதணும்னா கூட தமிழ்ல எழுதலாம். நான் தமிழ்ல பதில் சொல்றேன். No problem. Okay. Okay, if you don't have questions, it's fine. You can probably take a note of this websites. Yeah, you can reach out to me through my website also anytime, right? If you have other questions. Okay.
Sir?
I think that we have a question.
Yes, sir. We have questions.
Yeah, so Kaushali Amani asked me a question. So how does the probiotics alter the skin microbiome? Yeah, so okay, so this is okay. This is specifically skin microbiome. That's good. Okay, so skin microbiome, as I said, right? Healthy skin has a certain composition of the microbes. Let's say you have, let's say I showed you 3 microbes, right? Maybe I can go to that section. Then you can. Okay, so if you see this one, this particular figure, the blue one, this is actually called Cutibacterium acnes, and this one is Staphylococcus epidermidis, and this one is called Malassezia restricta. So generally in a human skin, what we are seeing is that this bacterium, Cutibacterium acnes, is actually the dominating one. And these two, let's say it's like 80 to 90%. And these two makes rest of it, like 15% or 20%. So this is what it looks like in a healthy skin. But in case of unhealthy skin, what we see is this one goes down and these two actually comes up. So it's probably something to do with the condition of the skin, or it can be something went wrong with these interactions that actually goes back. But if you apply the probiotics, for example, the Biomedical thing that I showed you, so this is the bacterium that you're applying to. So what you're doing is you're actually enriching that particular population. So then the other two goes down, right? So now you are rebalancing the system. So that's how it works. I hope I answered the question. Okay, Abhinaya Subramaniam, can you comment on how bacterial can be infection? Okay. Yes, people do consider it as an organ these days. So it's a little bit tricky, right? So I'll tell you. Microbiome is not only on the skin, right? So it is also present inside our alimentary canal. We have a microbiome. Even in lungs, people say there is a microbiome, and we have a separate microbiome, which is oral microbiome. These and the nasal microbiome. The skin microbiome is different from the nasal microbiome. This is different from the oral microbiome. This is different from the gut microbiome. And even within the skin, you have vaginal microbiome that is different from the rest of the skin parts, right? And especially for female, people, there is something called bacterial vaginosis, right? Where there is a irritating, it's a condition where it's a itchy condition. I think it's an irritating condition. But the thing is, from a microbiological perspective, what we see is for a healthy individual, there is a lot of lactobacillus, and the unhealthy individual who have this condition has less lactobacillus, and then there is a lot of anaerobic bacteria that is growing. So what we know now is that these microbes are very essential for our living, right? And if you basically take off everything, your immune system doesn't work nicely, and many of your digestive mechanisms don't work. And also, there is a lot of mechanisms that are reliant on these microbiome, let's say for preventing pathogens, etc. So we have now understood that these microbiomes are really important for our survival. That's why people are, considering this as an organ. And also, if it is just a bacteria from the environment, all the animals should have the same microbiome, but it is not like that. Humans have a different set of microbes. Let's say you take a dog, even though they are very close to the humans, they have a different set of microorganisms. So all these are very different, which means we are all evolving separately with these microorganisms. And if you think about a mitochondria, which is a cell organelle, mitochondria was once a bacteria, right? That was out there, and then it actually started collaborating with the archaea, which is also another microbe. They actually came together, and then the bacteria get inside archaea and then made a eukaryotic cell. So that's how the evolution happened, right? So in that sense, over time, because they are very much dependent on us and we are dependent on them, we can consider them as an organ, especially for us, right? I hope I was able to tell you. Okay, there are multiple questions here. I will try to go faster. Kirtana, please explain the top 3 innovative applications of microbes in technology. Okay, top 3. Microbes as okay. You probably have heard about FMT, which is fecal microbiome transplant. This is also something with the microbiome, which is so for one example is C. difficile, which is Clostridium difficile, which is an infection that happens in the gut. And antibiotic treatments won't actually help them because they are very much resistant to a lot of antibiotics. So fecal microbial transplant is something like, from the same household, they will take an individual's gut microbiome, closely related individual, and then they will replant the microbiome of that deceased person, and then this is actually changing the whole microbiome, and then they are fine. So that's a very well established and very well working clinical application. And then you can think about okay, so Craig Venter, right? So who sequenced the human genome first? So Craig Venter Institute, they are developing something called a synthetic cell. They have already developed actually. So what their idea is that a synthetic cell is a cell where, okay, what does a bacteria or a cell do? It has a lot of genes, and not a lot of genes are needed for its just a survival, right? So they are needed for, let's say, prevention from other organisms or harsh environments, etc. So they wanted to remove the genes that are not useful for just living, taking nutrients, dividing, only this. So they reduced all the extra stuff, and then they made it as a synthetic cell, which means they basically synthesize the whole genome chemically, and then they took an empty cell, they put the genome inside, and then the cell started dividing. So it's a synthetic cell. Why they're using this? Because it doesn't have a lot of other things. It can only do what it has to do, right? So you can put a gene which can produce a product, which can produce a drug, or or a fuel or something, and then this will efficiently produce that compared to other cells because they have a lot of luggage to carry, right? So that is another one. I hope you get that. So there are multiple things. Then I would recommend you to go look for it. And I would like to go further because there are other questions. But if you have things, like, you can contact me later. Harishant, Malassezia furfur is normal skin pathogen. I used these for my research. Okay, that's good. Yeah. Malassezia furfur. We are actually working on Malassezia restrictor. That is another one also. So basically, the Malassezia is not actually a pathogen. It is an opportunistic pathogen, right? So this is actually a part of a healthy human system, and at some point they become a pathogen because of the conditions that it is going on, which we are trying to also understand. Right? That's great. Okay, you're asking what you can do. Yeah, just write to me. We can discuss about what you have done so far, and then we can find out what you can do further. That'll be great. Yeah, I'll be happy to help you. Arunesh Prabhakaran, so can I get any info about enzyme assay and how and where to get internship for that? Okay, so there are two types of internships. One, paid internships, and second thing, free internships. Paid internships are a little bit hard to get, and it depends on if the industry is ready to do, right? I would suggest that go to a local university to start with. If you have your holidays and things like that, contact professors who are working on enzyme chemistry, biochemistry department, for example, and then write to them that you are interested in learning enzyme systems, and he will be happy to work in their lab. And you will not get paid, but this will be a very good thing because you will be attached to a PhD student or a postdoc in the lab, and you can tag along. You can learn a lot of things and so on. I would recommend doing that first. Okay. Kausalya again. Consortium of probiotics are used in application specific strains are used in okay. Right. Consortium is something that is very recently understood. So people traditionally work with single bacteria. Like for the Biomedical also, it's a single bacteria. The FMT, which is the fecal microbiome transplant that I told you, this is a consortium, right? And we know that the single microbes are very, very sensitive to a lot of environmental changes, but not the consortium. Consortium are very resistant to environmental changes, and they can actually, they can be very resilient and can be a very good therapeutic agents, right? So, but then the thing is, it's very, very infancy. Like for example, in Celsi, we are actually pushing a lot about consortium because in nature, if you see, there is no one place or very rarely you can see one single bacteria dominating our ecosystem. It's everywhere. It's a joint collaboration of a lot of different microbes. They are also specializes, specific microbes to specific functions, and they all together make the system work. So it's a very new thing, but that's, in my opinion, that's the future. So everyone is trying to make consortiums work. The problem is that how to understand that. So for example, in this case, this is only 3 species that we are trying to understand, and it's a relatively simple model. But in many cases, it's like hundreds of species which we don't know how they are working, right? And there is a lot of room for discovery there as well as innovation. Okay, moving on. Abhinaya, thank you so much for your explanation. Okay. Glad that it helped. Herbal drug study used. Okay, that's great. Yeah, Harishan, please do. You can check into drvideo.com, send me an email, or vidudalai@gmail.com, or contact your professors who contacted me. I'll be happy to have a call with you or something and find out how we can help you. Yeah. Kirtana. Okay. Okay, that's great. I'm happy that you guys liked it. Okay. I think that's all the questions we have. Okay. If that is all, I think we're good. I'll just put up the slide where you can find me.
Thank you, sir.
Yeah, no problem. Can we end here, or how do you want to proceed?
Thank you, sir, for your valuable speech and great knowledge.
Thank you. Can I leave now, or? Okay, all right, that's great. Yeah, it was nice talking to you guys. It's nice interacting with you. Thanks for all the questions, and thanks for inviting me. Yeah, good luck for everyone. Bye bye.
Thank you, participants.
If there is a magic on this planet, it is contained in a cup of coffee. We will meet after a quick break. The participants are requested to connect it back by 11:30 a.m. sharp. Thank you. By joining using the same link. Thank you.
ஹலோ ஹலோ
சார் கேக்குதாங்க சார்?
கேக்குது. உங்களுக்கு கேக்குதுங்களா?
எஸ் சார், எஸ் சார், கிளியரா கேக்குதுங்க சார்.
கிளியரா கேக்குதா? ஓகே ஓகே. அதான் செக் பண்ணிக்கலாம். ஏன்னா ஸ்டார்ட் ஆனதுக்கு அப்புறம் உங்களுக்கு செக் கேக்குமா கேட்காதான்னு தான் செக் பண்றதுக்கு கேட்டேன்.
ஓகே
கிளியரா
சார், கேக்குது சார்.
கேக்குதுங்களா?
ஆல்ரெடி செக் எஸ் சார், கிளியரா கேக்குதுங்க சார்.
ஓகே ஓகே ஓகே. சரி சரி ஓகே. சரி தேங்க்யூ
தேங்க்யூ
தேங்க்யூ.
சார். ஓகே சார், தேங்க்யூ. Sir, can I answer?
Yeah, hello.
Sir, shall we start, sir?
Oh, yeah, I'm ready.
So,
There's nothing better than expression in words. It's indeed a pleasure to welcome you all back to the second part of the session. Exceeding your own expectations is something that can take you to great heights.
Welcome to Impact Theory. Yeah, here, my friends, because you believe that human potential is nearly limitless, but you know that having potential is not the same as doing something with it. So, our goals with this session is to introduce you to the man full of ideas that will help you actually to execute on your dreams. All right, I'm profoundly delighted to take an opportunity to introduce the chief guest for the second session, who is going to talk to us about nanotechnology towards biomedical applications. This is a subject in which we all deeply get interested because over the few years, there has been a steadily growing interest in using nanomaterials in different biomedical applications and for health wellness.
On that note, on behalf of Department of Biotechnology, Ksrcas, let me introduce and welcome the guest speaker, Dr. Gopalu Karnakaran, who is a Brainpool Fellow and Research Professor at the Biosensor Research Institute in the Department of Fine Chemistry, Seoul, National University of Science, Technology, Republic of Korea. In the years 2011 to 2014, since he earned doctorate, he has published 96 articles on the subject he has to talk to us about today. He is the winner of Korean Research Fellowship grant under Ministry of Science and ICT and National Research Foundation of Korea. He has received 4 funded projects and successfully completed 3 among them. He is currently working on this project funded by Basic Science Research Program through the National Research Foundation of Korea funded by the Ministry of Education Korea. He is also a reviewer in about 30 international peer-reviewed journals. Dr. Gopala Karnakaran was invited to speak because he is an expert in nanotechnology. We were delighted with your expertise and knowledge in every possible way.
We are really thankful to him for taking the time out of his busy schedule to attend the function. So, without consuming any time, I would like to call upon Dr. Gopalu Karunagaran to enlighten us with the inspiring words. We hope that the students are able to seek a little guidance and imbibe something from his thoughts. We would be delighted to have your suggestions and questions at the end of the talk. Enjoy the event. Over to you, sir.
Thank you so much for the nice introduction about me. I'm very happy to be a part of this mega event, and here I'm here to talk about my presentation. So basically, this presentation I made in a simple way so that everybody can understand nanotechnology easily. So let's get started. So these are the contents of my presentations. So basically, I will give you some introduction, then history of nanotechnology, and then some ancient nanomaterials, and then features and properties, and then approaches of nanotechnology, applications of nanotechnology, and then biomedical applications, and future challenges in nanotechnology, then disadvantages, summary, and acknowledgement. So the introduction about the definition, the every topic have some definitions. So about nanotechnology, the branch of technology that deals with dimensions of less than 100 nanometer, especially the manipulation of individual atoms and molecules. When individual atoms and molecules are combined and form in 100 nanometer size, it is nothing but a nanotechnology. And that nanotechnology was coined by a person called Nario Taniguchi. He's an eminent scientist who works on nanotechnology. So he introduced the term nanotechnology. Nanotechnology mainly consists of the processing of separation and consolidation and deformation of material by one atom and with the other to form a molecules. So basically in nanotechnology, the small atoms are combined to form a bigger molecules, and in the size range of 100 nanometers. So whatever works on in between the nanometer scale, it comes under nanotechnology. So these are the definitions. So what is the history of nanotechnology, when it started? So generally, there is an eminent scientist called Richard Feynman. He is like the father of nanotechnology. So he was the first to introduce this type of concept in the year 1959 with a lecture about the nanotechnology, and he told that there are plenty of rooms at the bottom. Like in the bottom, there are a lot of rooms available, like that he introduced in his lecture. And he was got a Nobel Prize in the year 1965 for the quantum electrodynamics. So he works on this quantum technology, and he got the Nobel Prize. So as I told you, he is the father of nanotechnology. So this is the person, Nario Taniguchi, who has introduced the term as nanotechnology. So this term is being introduced by him. And then further, Professor Eric was the person in the year 1980 who introduced about molecular machinery, manufacturing, and computations. So why about this nanotechnology? So earlier, actually, this is not an new technology, new advanced technology. Everybody will say that nanotechnology is booming everywhere and like that, but actually it is being used in the previous history itself, but it was known now that it is being used. So around 1,000 years back, the gold and silver colloids are used for the glass painting. You can see here the glass painting is because of the gold nanoparticles being used. Different nanoparticles of gold have different colors. I will show you the next slides. And then for in the more than 2,000 years back, the sulfide nanocrystals are used for the dyeing of hairs. So dyeing of hairs is also a kind of nanotechnology application. And then Lacrosse cup. This cup was also a alloy of gold and silver. It will be having no color, like it will be greenish color um, when it is been without light. But when light is passed, it will show that this type of fluorescence color. So these are some kind of historical knowledge that already shows that nano materials are being used earlier also. So these are the timelines. I will show you that how the progress of nanotechnology is coming from the earlier to now. So it is like earlier, during first generation, small aerosols, colloids, coating, and then polymers were used for different kinds of applications, which was started from 2000. And then in 2005, the active nanostructures like bioactive materials, health affecting material, targeting materials have been introduced with the active nanostructures. Then the systematic system was introduced, and then the third generation was from 2010, in which the 3D printing and then the architecture designing of this nanoparticle has been increased. And then during the 2015 till today, the molecular machinery and the nanocrystals have been generated every day in a regular scale for the different wide applications. So I will tell you briefly in the next slides. So what is the features and properties? So before we start to know more details, we should know what this nano and how it is important, and what are the basic concept of this nano materials and all. So um, basically, the particle size, the size of the particle is very important. Like everybody knows that earlier there was a very big room of systems as a computer, but as a day changes, we in the phone we can use like a computer. So this advancement, the bigger size to convert it into nano size, it is like what it is happening. So the nano size is being introduced regularly to to reduce the space and to do the advancement in a very fast way. So as per the particles are small, high surface to volume ratio will be increased, react differently, and then act differently. New properties will be introduced as the size variation is available. There will be a lot of different properties will be present. So and then it will interact with the light differently. I will tell you how with some examples that how the nanoparticles are interact with light differently, how it appears differently. So are on the scale of small biological structures also, these nano particles plays an important role. I will tell you more details in the coming slides. And then quantum mechanics meets classical mechanics, and interesting new structures will be produced, and interesting material with nanoparticles embedded will be also generated. So basically, if you see, like in life science, we know that these are the material which have the nanoparticles. Like DNA is having a size of 2 nanometer, and then the cell wall is made up of from 5 nanometer thickness, and then these are the molecular mechanics which have the 10 nanometer diameter. If you see the blood is around 5,000 nanometer diameter, and the virus are around 50 to 100 nanometer scale. So basically, nanotechnology is not new. This is just the basic thing which everybody knows already, but we are not aware of what it is exactly. So if you understand more easily, you can see here that ant is having a around 5 micrometer size, and then when the size is reducing, these are the, you see here, these are the in micrometer scale. Then blood is also still in the micrometer scale, and then when it goes to till DNA, the size is around 2 nanometer. So and everybody knows that when the DNA molecules interact and they move faster and they store more information. So uh, like in biotechnology, we also heard that there are a sequence of genes encoded having a lot of informations. So these nano particle is like we can use DNA also as a nanoparticle, which can be used for further applications. So there is a correlation between biotechnology and nanotechnology. So nanotechnology deals with the nanometer range scale. So anything, whether it is like a material or a biological material which comes under the nano range, it comes as a nanotechnology. So now I think you will be more familiar that everything is nano, and we already know it. So I will give you some more examples from the nature, like how this nanomaterial have a very wide application and how it is been very important. Like if you see the beetle's color, actually the beetle itself have some type of nano crystals uh, present in their surface, and which is having 400 nanometer scale range. So what happens when the light enters to that hit to the surface? Actually whatever we see, it is a reflection of light. It is a reflection of light. Like the light is composed of around 7 colors, about from Vibgyor. So basically, when the lower nanometer scale range is being reflected, for example, to understand more easily, for example, if you see a tomato, so tomato is red in color, right? Everybody knows why it is red color, because all the 7 colors, when the white light hits to the pigments of the tomato, the other 6 colors are reflected, are like dispersed, and only the red color only is revert back to our eyes. So that's why we see the tomato as a red color. Similarly, this beetle, beetle have some kind of material which is around 400 nanometer scale. So when the white light enters there, the only that violet like kind of colors will be reflected back. So that's why we see only one color kind of thing. So in similarly, when you see the peacock also have different kind of patterned nanoparticles over the surface. So whenever the light enters, and then only specific light is reflected back to our eyes, we observe that this is the color, like it's a rainbow kind of color. We cannot see like a specific color, but with a range of colors, because of the surface have different kind of nano structures embedded on the surface. Similarly, you can see the gacho that the you can see like lizard and other thing and all. These have the property of like attaching in the wall and just move on. That movement is also related with the nanoparticles, because the small nanocrystals are present on the surface of the toe, and then it helps in the binding of it. I will show you in the next slide. You can see here gecko feet has the you see, you know already this gecko feet. And if you go in depth, you can see that these structures are a form of cetal array, and then this ceta is composed of 100 nanometer material. So these material actually helps in the adhesion, and a very good material to hold on the surface of the wall. So these are behind the nanotechnology which is present in already in nature. So in nature, as I told you, that there are some kind of optical properties also, like in basically wherever you go to spectroscopy studies and all, you know that light is a white in color, but having different kind of ranges, like violet to, we always represent like vibgyor. So vibgyor, so from violet, blue, green, yellow, orange, and red color, these are the colors based on the wavelength which is present. So to understand more easily, like it is made up of particle physics. Like if you go to the theoretical way, so whenever you know that from the sun there are lights are coming, that are everywhere, the lights are being the particles are moving, they always have a particle nature and the wave nature. So when the particle, whatever we see and observe, everything, if you see in the molecular level, it is formed or happening in the atomic level. So atomic level, when this white light will be composed of these different patterns of the nanometer material, and these nanomaterial material will be reflect whenever the other colors are like dispersed. So you cannot see that color. The only color which is revert back, that color only we can observe. So generally if you have color blindness, you cannot see the specific kind of color because of the wavelength difference. So to more details, like if you see from 300 to 435, there is violet color, until the red 740 nanometer to say like 625, it is red color. So these are, you can see, these are nanometer scale having wave number. So why I have told these things, because you can see here the particles size plays an important role for their properties. Like you can see here, silver nanoprism have 100 nanometer, and its colors is red color. You have seen in silver, when the silver is like silvery in color, right? But when the size are reduced, the color is changed. So this is what the property difference because of the size. Similarly, if you see golden color of like this, it is 100 nanometer, but when it comes to sphere of 50 nanometer, it will change to like a green color. I will show you those. So the size of the particle plays an important role for the variation in the properties. To make you more understand, if you see here, this is the gold nanoparticle which is having 10 nanometer scale, and when it goes to 100 nanometer, the color is different. You can see the color is changing because of the size of the nanoparticles. And when it goes beyond this color, So like if it is like 1 micrometer and like more and more, we will see the gold as a golden color. So when it reduced to, if it comes back to 10 nanometer, this is the gold color in 10 nanometer. Similarly, here also you can see the quantum material. When it comes with a higher size, it will be red color till to like violet color. So as you have seen in this wavelength also. So here when it is bigger size, so it shows different color, red color. When it is lower size, it shows like a bluish color. You can see here. See, to understand the light property based on the size. So this is why the particles have so much variations. So in this way, it is having a very variation in the properties also. So there are different various approaches of nanotechnology in which how these nanomaterials have been produced. So basically, there are only two methods. One is like top down method. The another is bottom up method. So what it is, if there is a like a bulk material, for example, you have taken a big stone, and when you cut and into small particle, when it comes to the nanometer scale, then it comes as a nanoparticle. So this is from top to down approach. So in this method also you can produce any kind of material whatever you want. The another method is bottom up method. It means you have to go to the atomic level. You have to take the precursors like small molecular atomic molecules, and then these atoms are combined to form a bigger molecules around 100 nanometer range. It forms a nanoparticles like. So that's why it is called bottom up approach. So from atomic to higher, and there, whereas top to bottom means the bigger molecule to smaller molecules. Only these two approach are being used for the different designing, synthesis, fabrication, anything about to produce nanoparticles. So now in nanomaterials, there are different classifications like 0-dimensional, 1-dimensional, 2-dimensional, 3-dimensional. So generally the gold nanoparticles, silver nanoparticles, all are comes under the 0-dimensional particles, whereas carbon nanotubes have a wide range of single dimensional. So they will be having a various length of nanoparticles. So that is called 1-dimensional nanoparticle. Then in 2-dimensional, now you have seen a lot of solar cells, solar panels are being produced. They are a thin film of 2-dimensional nanoparticles. So generally nowadays, solar cell, you have seen a lot of places like solar panel is produced. So what they are doing in the panel, they are coating with small nanoparticles in the 2-dimensional range, so that it can like help in the formation of solar energy. And then the 3-dimensional, in this 3-dimensional you can design and you can develop any material in the form of bulk material. So you have to design, like if you can code on the drug also. You can design by adding some molecules, different molecules, or some receptor proteins, anything you can design in the 3-dimensional structures. So now comes about what are the applications of nanotechnology. So uh, there is no like area where the nanotechnology is not being covered. Everywhere now nanotechnology is already covered all the area, and it is present everywhere. So now, like for example, energy like Tesla and other, there are a lot of companies which are now using nanotechnology advancement for their energy applications. Like you can see in energy, solar cell panels, like batteries and other thing, everywhere now nanotechnology have important role for the advancement. And then medicals and drugs, like for drug designing and all, nanotechnology works. Nanobiotechnology, like with molecular mechanisms and all, it is been using. Nano device are being produced. Then chemical engineering is being done. And then defense and security also nanotechnology is working. And then bioengineering also nanotechnology is taking part. And then in cosmetics also, the nanoparticles are being used for the sunscreen and applications. And then nanofabrics, so nanofabrics also being used for the like there are nowadays thermocouple fabrics. So if you go just outside and you can charge with your clothes directly and to your phone, and you can use it. Like now these type of advancement are coming day by day. So now I just focused on only one area, like biomedical applications, because you are all are from some kind of bio related field. So this will be more useful for you to understand how these biomaterials are being used. So to make you more familiar, already you know all these things, but still I just want to make you more familiar. Like if you see the cells, the vesicles, cytosol, centrioles, mitochondria, if you go to the size of this material, you will come to know that these are all nanoparticles. These are all kind of a nano range, but you know that how these are functioning, even though it is a size of like some nanometer range, but they have a specific and important application and use for in a different way. So these all are nanotechnology only. So these are all nanomaterials. So in future also you can use like this Golgi body for some kind of applications, like mitochondria also for some kind of application, ribosomes also being used for some application. You can take each and every material as a nano material for different kind of applications. And to make you more familiar, like this virus now, coronavirus and other viruses, all are also a part of nanomaterials being embedded, and they are doing a lot of advancement and things to the environment. So now you understand what is nano already, that what is nano and what are the materials and all, how you produce and all. Now it comes like how it is applied. You can see here, these are the nanoparticles, nanocrystals bound with a specific site to show the presence of the cancerous material. Like earlier we used some chemicals and all, but now the chemicals are being replaced by use of nanomaterial, nanocrystals, for the detection and for to labeling of some fluorescent material, so that it can go and target the specific place and to show the place of targeted cancer. So in order to understand the type of cancer and the range of size, so that it can be treated easily. So in the tissue engineering also, now nanoparticles being used with different gold nanocolloids, silver colloids and all in three-dimensional way, so that the wound can be healed faster. So that generally if you see there is some wound, so we just apply some type of chemicals. That chemicals can be replaced with these types of gold or silver colloids, so that it can be recovered fastly. So it will help in the tissue engineering. And then detection of chemicals and war weapons or bioagents can be detected from the use of nanoparticles. So if you have some nanoparticles being embedded on the surface, it can be easy for it to detect any kind of biological war weapons or any chemicals which are harmful by the same like concept of enzyme and antigen binding mechanisms. It will give some signals to detect the chemicals and biological warfare agents. So in future, nanotechnology is working on the formation of artificial red blood cells. So what's its role is, it will be spare of from 1 micrometer diameter to 1,000 atomic pressure vessels, and then it will have the power to produce and to eradicate all the unwanted things, and then it will produce more and more oxygen to the system by naturally. So these are the advancement are planning in the field of nanotechnology towards biomedical application. Then fixing of damaged bloods. So generally nanoparticles are being introduced in the blood system, so that it can repair all the blood cells if it is being damaged. Then nanotechnology in the drug. So generally nanoparticles are being coated with some kind of carriers or agents, and it will helps in the movement of these molecules towards the specific target site. So it provides new option for drug delivery and drug therapeutics, enable drug to be delivered to the right location, and predetermined schedule for the optimal treatment, attach the drug to the nano size carrier, become the localization at the disease site and cancer tumor. They release medicine that kills the tumor, and currently treatment is through radiotherapy and chemotherapy, but this can be replaced with these type of nano carriers. And nano robots can clear the blockage in the arteries. So these are the way how these nanoparticles can be used for as a drug. So in future, as I told you, these nanoparticles can be used for different kind of robotic. So nano robots as imaginary, but nano size delivery system could break apart kidney stones. Even if you have a kidney stone, this nanoparticle will be entered, and then it will break the kidney stones, and it can remove all the toxins and clear plaque from the blood vessels and ferry during drug to tumor cells. So it will be act as a ferry. It will go and move the drug to the tumor cells also. So, and in future also, this nanotechnology is being will be used for the nerve cells, the possible brain to brain neurocomputation and prosthetic biosensors. These nanoparticles can be used. Then, as I told you, for drug like disease detection, we can use different kind of quantum dots. So these are very low materials which will have very low effect. Whereas if the already existing chemicals which will be introduced, we have to use in the large amount. But the quantum dots will be very low quantity, but it will go and target the specific place, and it will give more results. So in it is in clarity, it will help in the lower of toxicity, and but it will give more efficient result for easy detections of these type of errors. So here you can see the tumor is been detected in three different places by using these quantum nanomaterials. So even in the low quantity, we will get the higher result and higher approximate result, and we can easily detect these are the place where the cancers are present, and we can easily treat it into the specific targeting place. And then growing nanofibers will help in the growth of heart muscles. So sometimes there is like some kinds of hole is being produced in the heart. So these types of nanofibers what will happen, it will be attached to the place, and it will help in the growing of the cells, and it will cover all the holes, and the heart will be functioning normally. So these are the nanofibers applications. Then as you already aware that everywhere is virus now. So this virus can be coated with the gold or silver kind of coating, so that it cannot attach, or it cannot harm other people. So this coating also will help to prevent the virus infection to us. And about the DNA chips, everybody, I think, may be familiar. Just to introduce again, like DNA and RNA molecules will be used for coding and to form a chip, so that the lot of informations can be coded and it can be used for various applications. So this use of DNA and RNA molecules also be considered as a nanoparticles applications. So for bio, as I told you, in biosensor also, DNA molecules attach the end of vertical carbon nanotubes that are grown on a silicon chip. These detect specific type of DNA in an analyte. So you can analyze any kind of gene coding and all by the use of these kind of biosensors. So now coming to in India, like nanotechnology been growing now rapidly in India also. Like IIT Mumbai was the premier organization in the field of nanotechnology. Then research in the field of health, environment, and medicines are still on. Starting 2001, the Government of India launched the Nano Science and Technology Initiative. And then in 2007, the Nanoscience and Technology Mission 2007 was introduced with an allocation of rupees 1000 crores for a period of 5 years. The main objective of these nano missions are basic science research promotion, infrastructure development for carrying out front-ranking research, development of nanotechnology and their applications, human resource development, and international collaborations. So, generally, this nano mission in the Department of Science and Technology is working still now, and they are over the period of time they are enhancing their funding for the research towards the nanotechnology. So, nanotechnology in India is also not like old; it is now is booming and been regularly been promoted for the next level of to match with the international level. Our India also been introducing these technologies. So about to know more about the uh, uses of nanoparticles, you can see here there are different nano industries are also emerged. So over the period of time, by knowing the advancement and utilization of these nanomaterials, there are different industries now working on it. So like equipment center, imago instrument, atom, hystronic ink, thermoelectronics, then advanced material like Cayman and nanotechnology, electronics, Intel, HP, Motorola, IBM, all are coming up. And then in energy center, nanodynamic, Carnuco and Simber, these are all the like energy sector. So you can see here there are like lot of industries are coming up to use the nano approach for their real applications. So, but what are the future challenge in nanotechnology? Still is had to be eradicate is reducing the cost of material and device, improving reliability, implication of nanotechnology to on society, and then environmental problems can be solved, innovation of nano drug which will be less side effect and more effective. So these are the area where we in still more challenging have to be eradicated. But what are the disadvantages? So we have seen a lot of advantages about nanotechnology, but now there is some type of disadvantage also. The engineering robot will perform jobs instead of people, which will result in a loss of jobs. So if more a lot of work can be done by a lot of people, but if a small particle or small robot will do all such kinds, so there will be a lot of loss of jobs for the people. There is first disadvantage. And then the waste related while making the material for nanoparticles are released into the atmosphere and can even penetrate human and animal cells and affect their performance. So whenever we have to work, we have to be very careful to release the nanomaterials towards the environment. So we have to be very careful, like some materials will be affecting, some material will not be, so we have to recycle or we have to dispose it in a proper way. Then agricultural countries will lose their income as a nanotechnology will take over it, and it is a very highly initial investment cost along with high manufacturer cost. So initially the initial startup will be very expensive. So these are the disadvantages. If any damage is done in a molecular level, so it will not be reverted back. So because all nanoparticles are working on the nano scale range, so anything you defect in the nano range, it cannot be reversed back. So it is very tedious. Like if it is in bulk range, you can just modify and anything, but whenever there is in our nano range, if there is any defects, so it will be a very like it will not be revert back. So these are the disadvantages. So in summary, nanotechnology, what is nanotechnology? It is an emerging interdisciplinary science integrated with chemistry, physics, biology, material engineering, earth science, and computer science. All of it, when it is combined, it is forms a nanotechnology. So, what is behind this? The power to collect data, manipulation, and particles at such a tiny scale will lead to new area of research and technology designing, better understanding of matter and interactions, new way to tackle important problems in healthcare, energy, environment, and technology, and a practical application now, but more years or decades away to be prolonged by the use of nanotechnology. So these are the summary. So these are the links to know more about this nanotechnology, like that NNNI link is available, NNI and link available, MRS available, Nano Hub is there, conference NSTI, UMI. These are the places where you can see about the conference on nanotechnology. NanoRite Center, these are the center where you can see the centers available who are working on it. And Nano in the News, these are the websites where you can learn more about nanotechnology and their advancement in the coming years. And last but not least, I would like to thank one and all present here, and I would like to thank to the chairman, to the manager, and to the all the head and staffs or principal, and to the organizing committee, and to the head, all the head of the departments, and all of you, and all to my guru and to my parents, everyone. I would like to thank one and all for giving me this opportunity to be a part of this presentation. Thank you. If you have now, the session is open for the discussion. If in the audience, if you have any queries related to or unrelated to also in this field, you can ask me. So now the session is open for the discussion. If you have any queries, please ask me. I would like to answer all the questions.
The session is now open for discussion. Participant can post your queries in the chat box.
Yes, you can ask me. So whatever doubts you have, you can just text me. I will answer you one by one. So there, I have seen that Kamali Kandasamy has just posted one message like, "Sir, I have many doubts in nanotechnology." So, what doubt you have, you can just message me in the chat box. Can you send me some more if you have any doubts?
Sir, I think they are typing their questions, sir. So they wait for some time.
Oh, okay. Thank you. So I received one question about from uh, Dharani Ramasamy, like, "Sir, please tell me some points about copper nanoparticles." So as I told you, copper, like you can you take a copper wire and you can just dilute with some chemicals and all. You will see there will be some colloids and all. So that copper will be converted into nanoforms, and that nano means like in the size of you are reducing it. You are taking a copper wire and you are just reducing it, and it will be converted into nanoparticles, and that nanoparticle can be used for different kind of application, like copper or very useful for biomedical applications. Like it can be used as an antibacterial material. So if you have some kind of infections or anything, you just apply the copper colloid over the surface of the some infections. So if you have bacterial infection, or if you have some kind of fungal infection in like in the body also, you can apply this type of copper colloids. So these colloids are nothing but nanoparticles. So as I told you, from top to down, so you are taking a bigger material and now you are reducing it to small particles in the form of copper nanoparticles, and you can use it. I hope it will be okay to understand it, right? Any other question you can ask me? So no more questions, I think. So it's okay, and now I conclude. I thank to one and all present here, and thank you for giving me this opportunity by the organizing committee and all, and thank you for inviting. I'm very happy to be a part of this event, and thank you one and all. Have a nice day. Take care. Bye. Good day.
Thank you, sir. Thank you for wonderful speech, sir.
Thank you, sir. Success isn't just about what you accomplish in your life. It is about what you inspires others to do. I would like to introduce our keynote speaker of today's session, Dr. Senthil Kumar Balakrishnan, who is the director of Microtech Laboratories, Adur. He has completed his graduation in University of Madras and completed his PhD in biotechnology from Periyar University in 2009. He was awarded for inviting renowned professors from foreign universities by Government of Tamil Nadu and received travel grant through Bharathasan University, Tiruchirappalli. He is a responsible member at Department Graduate Council, Haramaya University, Ethiopia. He published about 63 research articles and he is also a reviewer in most of the international journals, including Springer and Elsevier. He is a lifetime member in Indian Immunological Society and International Society of Infectious Diseases. He is a potential person who received several research grants and he has published books on industrial applications of microbial enzymes and practical immunology. Without further ado, let us welcome Dr. Senthil Kumar Balakrishnan to present a lecture on probiotics for human health. Over to you, sir.
Okay. Good morning, all of you. So I am very happy today uh, to be the speaker in the international conference, and it is a coincidence that today is the World Health Day, and the topic which I am going to present is also related to this human health. So the topic entitled "The Probiotics for Human Health," which is likely to be presented in this session. So, as a human being, we must know well what are the health benefits based on certain foods, so that we can lead a healthy life. So, based on this background, I have chosen this topic. So, many of you might aware of probiotics, and some of you may not aware of the probiotics. Actually, the term probiotics means pro means promoting and biotic means this is general meaning. Actually, the probiotics is not new for us. Even with the history of man, when the human beings started use cheese and fermented milk, they started using probiotics. Here, they have no idea about probiotics, whether it is probiotics, but they blindly knows something that if they consume this cheese and fermented milk, they get health benefits. Then, Eddie McKinnicoff, who described about the probiotics early in the 20th century, actually, this is also based on his native observation. The dwellers in the Bulgaria, they living for a long time, more than 100, something like that. So from that he got something. So since they consuming more probiotics, and that's prolong the life of the people. In 1960s, Lilly and Stilwell coined the term probiotics. So for the first time, the term coined by this Lilly and Stilwell in 1960s only. Then proper definition is given by WHO in 2001. Actually, what is the proper definition for probiotics? When we take living microorganisms in the adequate amount that would confer the health benefits, this is what called probiotics, and also what is defined by World Health Organization. Probiotic candidates. Actually, the probiotics are microorganisms. It may be of bacteria, fungi, or yeast. Of these, bacteria and yeast playing vital role. So, even though n number of probiotic candidates are available, even in the market or in the different food sources, however, very few are very important, like Lactobacillus species, Bifidobacterium, then Lichnostock and Saccharomyces. These are very important. So in the day-to-day life, we may consume this knowingly or unknowingly along with other foods. So there are several species within the genus that are also can be used as probiotic candidates. So including Streptococcus, Bacillus species, Staphylococcus, and Enterobacter, Aspergillus, all these can be used as the probiotic candidates. Then probiotics rich foods. So naturally, certain foods are rich in probiotics. So, for example, buttermilk, or curd, or cultured milk, or idli dosa, and kefir, and miso, organic miso, pickles, and rice. So old rice soaked in the water with a little bit curd or buttermilk. So that would be a good probiotic rich food. So in the day life, many of the people, especially people living in the rural area, they do consume more amount of that old rice soaked in the water. So they do cook in the evening, and the left out rice will be soaked in the rice. In the morning, they will consume it. So that will be a good one. So there is more health benefits if we consume that old rice that is soaked in the water. So if we need, we may add some more buttermilk or curd, and also along with onion, something like that. So even idli is the best healthy food which is recognized by World Health Organization. So they recognize the idli as one of the health food. So if we are a patient, we may be asked to have idlis something. Sometimes depends upon some infection, they may ask us to take buttermilk with rice. So based on that requirement, we may consume. But in the day-to-day life, we may consume the idli, dosa, curd, buttermilk, or pickles. Different types of pickles are available, as you know. So these are probiotic rich foods. So even we are having many states in our country. So everywhere the cultural differences will be there. So each and every place they may have some different food habits, and they may have some fermented foods. Even likewise in other countries also. So for example, if you take China or Africa, so they may have different fermented foods. So all those are rich in probiotics. So where they are habitating, accordingly they are consuming and they are getting their health benefits. Characteristics of probiotics. So what is the characteristics probiotics that should carry? So that is very important thing. So since it is a microorganism, when you say microorganisms, it is not all harmful. So some are beneficial, some are harmful. So here we are talking about beneficial microorganisms. So if we want to use the probiotics, so that should derived or isolated from the human origin. Why? Because even the probiotics is not only meant for humans, even it is meant for animals and also for even fishes and others. So, for example, even I know one of my friend who is having a poultry farm in Ethiopia. Actually, he is purchasing the probiotics from the commercially available companies, and he is propagating it, and he is giving along with the feeds to the chicks. So the chicks are very healthy. So likewise, many of the probiotics are available. So even the human-based probiotics may be given to birds or animals, but for human beings usage, it is better to give the human origin probiotics. Why? Because the systems may be different. For example, if you take animals like cow or ox, actually their digestion pattern will be different. Actually, they consume mostly of cellulose, rich materials, so it is not easily consumed or digested by them. Why? Because the animals themselves, they cannot produce the enzymes that is required for the digestion of the cellulose. So however, the microorganisms inhabited in their digestive part is playing vital role. So because of that, uh, it is quite different for humans and animals. So it is mandatory for the human usage. If you want to commercialize the probiotics, it should be originated from the human, and it should have the property of alpha hemolysis, sorry, gamma hemolysis. Actually, the hemolysis is the pattern which is useful for differentiation of certain medically important bacteria, and it is also one of the point that is to differentiate the pathogen and others. Here, there are three types of hemolysis: are there alpha, beta, gamma. So alpha is partial hemolysis, beta is complete hemolysis, whereas gamma is non-hemolysis. So if the strain is having the property of probiotics, but it is having the beta hemolysis, means that is very dangerous. Upon the conception, it may start to lyse our heart disease. So that leads to anemia. So that is not to be allowed. So the strain should have the characteristics of gamma hemolysis, and they should be sensitive to antibiotics. Why? Because if it is resistant, then it is difficult. If it is sensitive, then no issue. Why? Because if they are drug resistant, they carry the drug resistant genes. There is a possible to transfer the drug resistant gene to other normal microflora organisms. Even we can say the probiotics might be many of the times part of normal microflora. So even I have published one paper in that I stated if the normal microflora organisms assuming or conferring with drug resistance, then it is very difficult. But because all the pathogenic organisms also may occur from the normal microflora, so then transfer of normal microflora and the normal microflora also possible, then it is very difficult. You cannot treat the person for any type of disease. So that much dangerous that one is. So if it is sensitive to antibiotics, no problem. So even if they are removed off, then again we can take in the form of probiotics along with the food, and again we can rehabilitate it. Then they should be resistant to low pH and the bile salt. Actually, these are the environment prevailing in the stomach and the intestinal area. Why? Because stomach secretes acids, so the pH of the stomach will be less than 4. So it should be tolerable. Otherwise, unless otherwise, they may die off. So while consumption, it may die, or it may be subjected to killed. Therefore, it should resist low pH as well as bile salt tolerance. Bile is secreted more in the intestinal area, so it should be resistant to the bile salt also. So another is resistant to digestive enzymes. So it is by nature many of the digestive enzymes like lipases or proteases like that it is secreted. Actually, they will even denature the appropriate proteins or lipids. If the organism which is being used as a probiotic is must be resistant to digestive enzymes. If they are sensitive, means they will be killed and they have no role to play there. So they should be resistant to digestive enzymes. And good volatile fatty acid producer, actually uh, on fermentation or other metabolic pathways, they should be able to produce volatile fatty acids, or we can say short chain fatty acids like propionic acid or butyric acid, something like that. Why? Because these volatile fatty acids will be source of carbon for other normal microflora microorganisms. So this is one of the important characters. Then it should have pathogen inhibition potential. That means it should have antimicrobial peptides. It should produce antimicrobial peptides like bacteriocins or others, or bacteriocin-like materials that should be produced by the probiotics is useful. Then another important point is they should be adherent to the intestinal cells and also mucosal cells, and they should not be non-cytotoxic. They should not perform any type of toxic, cell damaging activities. They should have broad metabolic spectrum, so then it is useful for us. So overall, it should be proved to be safe clinically, even in the animal trials, human trials. It should be proved. Then it is permitted to use as a probiotics if it is to be commercialized means. So overall, it should be safe to use. Then only it can be accepted as probiotics. So there is a term GRS, generally accepted, um, uh, generally recognized as safe by US. So likewise, it should be accepted after that animal trials and all. So then the new strains of probiotics can be used for human usage. Likewise, the probiotics is available for even animals also. So animals, birds, which is rearing for the meat purposes, they can be given with the probiotics. So then we can enjoy with that. So another is prebiotics and its sources. So probiotics is important. At the same time, prebiotics also important, because prebiotics are the source of nutrition for probiotics. So if the prebiotics are more, then there is a possible for more improvement of or inhibition of probiotics. So there are 12 healthy foods high in prebiotics has been reported. Even tomatoes, artichokes, onions, garlic, chicory, dandelion greens, asparagus, leeks, berries, bananas, flaxseed, legumes. These are certain. And prebiotics, why these can be a prebiotics? So due to its chemical composition, like the fructans, fructooligosaccharides, galactooligosaccharides, inulin, lactulose, soybean oligosaccharide, and leavens, peptic, gyro, phyto, like this. These are the compounds present in that foods that makes it as prebiotics. So accordingly, these uh, compounds are present in this onion, banana, etc., something like that. So here, the milk is very important one. Why? Because the newborn babies up to 1 year or up to 2 years, it is given with mother's milk, and after that, cow's milk. So that is due to the presence of oligosaccharides. That is essential for the children's growth. So likewise, now and then it is necessary to use appropriate vegetables, fruits, and others, legumes, seeds, uh, so then we can have prebiotics, so that will enhance the growth and inhibition of the probiotics. So the difference between the prebiotics and probiotics is prebiotics are non-living, while probiotics are living one. It is non-digestible, and the probiotics that can enters into the intestine from the ingested foods or supplements. Uh, the prebiotics keep stable, while the probiotics can be filled or increased temperature. It will be filled. Then prebiotics are not affected by stomach acids, digestive enzymes. However, probiotics also not affected, but up to some extent it can withstand. Uh, and the prebiotics are stable, and they are not deteriorate when stored for a long time. But probiotics have the shelf life. So improper storage that leads to destroying or killing the cells. So likewise, prebiotics and probiotics can be differentiated. So there is a relationship between the normal microflora and probiotics always. Here, the important thing is when we born, from the birth time onwards, we acquire the microorganisms. Actually, uh, it is impossible that life without microorganisms. So microorganisms are also part of our body, even it is established. So when you say normal microflora, it is the microorganisms or microbiomes inhabited in our body. So in the different track system, it is existing. So when we start acquire the microorganisms, so until birth, the fetus is free of microbes. During the delivery time, it is acquiring the microorganisms from its mother, from through pass through the birth canal, and after that from the environment, yeah, and then along with the water, milk, other foods, the child starts acquiring the microorganisms. So it may be gastrointestinal tract, urogenital tract, skin, and oral nasopharynx cavity. So these are the systems that are inhabited with the microorganisms. Actually, uh, in 1894, Rnas developed a system to rear the notobiotic animal, so-called germ-free equipment. He said, actually before that, Louis Pasteur also did not believe that my human beings cannot live without microorganisms. So they did the experiment on rearing the notobiotic animal, and notobiotic animal is nothing but germ-free animal. So what they did is they dissected the pregnant rabbit and they obtained in the aseptic condition. Why? Because if it is giving natural birth, it may be acquiring some microorganisms. In order to avoid that, they take the pregnant animal and subject to cesarean in the aseptic condition, and they take the small one and they kept in the notobiotic equipment or cage, so where the sterile environment will be provided, even water, food or feeds, anything should be provided in the sterile condition. So they react and then they come back to work. Actually, for healthy life, our intestine must be developed. So, for example, if the notobiotic animal have lean and thin intestine, while the normal animal have developed intestine. Why? Because whatever you eat, the absorption takes place through the intestine. So unless the intestine is developed one, the absorption capacity will be less, so that you will not get energy and the growth will be affected definitely. So likewise, the notobiotic animal and the normal animal can be differentiated. So notobiotic animal have uh, less immunity, and its immunity is not developed one when compared to the normal animal. And the notobiotic animal also requires more amount of vitamin B and K. And notobiotic animal will not suffer from dental caries. Why? Because there is no normal microflora. So there is no way for dental caries in those animals. So they already told, as I already told, they present with thin and lean intestine. It is not underdeveloped one. So likewise, so if any person is fat, we cannot say the person is healthy. So his intestine is well developed along with that normal microflora with the probiotics, then it is well and good. So his system will be function normally, and he can live a healthy life. Unless otherwise, it is very difficult. So opportunities for microflora modulations after the birth, like placental factors, neonatal factors, postnatal factors, these are, and the placenta actually during the gestation period, all the nutrients is supplied through the placenta. So during the delivery time, mode of delivery and gestational age. Actually, one thing, the child intelligence is determined by uh, maternal only, not by parental. Why? Because that is through the microflora. If the microflora is acquired by the child from its mother properly, means its intelligence will be good. So otherwise, uh, its intelligence will be affected. Uh, then postnatal factors like whether it is feeding with breast milk or formula milk, and the location of the living family members, host interactions, and the maternal diet. The maternal diet also one of the important factors. All because if the mother is healthy, then the child also will be healthy. Otherwise, it is difficult. Then weaning. Then during the toddler and the childhood days, likewise from the gestation period onwards until the toddler childhood stage, the microflora will be modulated. So if the child is given with the proper care, then there is no issue. And if the child is not given proper care, means then it is difficult. Its microflora is affected, means its growth and other intellectuals will be affected definitely. So if the child is born by vaginal delivery or cesarean delivery, that makes a difference. Actually, vaginal delivery is the normal, and the cesarean delivery is we have to do artificially. So then accordingly, the change of microorganisms should be there in the child. So some microorganisms may be enhanced, some may be diminished. Even the diversity, the local and regional diversity, so-called beta diversity, will be affected. And in case of breast milk and formula milk, actually, nowadays the formula milk also given along with the probiotics nowadays. So there would be some different or known few probiotic strains will be there in the formula milk. So that is alpha diversity. So likely the child should acquire the microorganisms, then they become into the normal microflora through different modes, and differences might be found whether it is mode of delivery or feeding with mother's milk or formula milk. So this is a source of bacteria in the human colostrum and milk. So from the maternal intestine, from the mother's intestine, the microorganisms that migrates to the mammary gland through enteromammary pathway. So when woman becomes pregnant, the glands will be activated, so start to secrete the milk. So during when the woman becomes pregnant, some pressure and physiological changes will be there. So then the normal microflora microorganisms from the mother's intestine to mammary gland will migrate. So when child feds with the mother's milk, it will acquire the microorganisms. Even the skin microflora also will be acquired by the child, and other environment and through other process also that is possible to acquire the microorganisms by the child. So these are certain factors that influence in gut microflora. So the gut microflora is very much essential. So external influences like diet, probiotics, prebiotics, antibiotic usage, lifestyle, any illness, and the living environment. All this determines the presence of gut microflora. So, for example, if the person is a patient and he is taking more antibiotics, then definitely there will be imbalance in the gut flora will be there. So, the density, diversity, activity of the gut microflora is very much essential. If it is normal, then it results in health. If there is any imbalance in the normal microflora of the gut, that results in disease. So called dysbiosis, the imbalance of normal microflora. Then internal host factors includes age, genetic stress, physiological processes, anatomical structure, and the physiology of the digestive tract. All these determines that gut flora. So these are varying depending upon the individuals. For example, one person at the same age, he may have healthy microflora, and another one person may be suffered because imbalanced normal microflora, especially that gut microflora. So that is it. So likewise, these external influences and the internal factors that determines whether the person runs healthy or suffers from disease. So the functions of microflora, we have three main functions: protective function, sexual function, and the metabolic functions. So here, protective function is if the normal microflora presence, including the probiotics, the pathogen displacement. Actually, the pathogenic organisms not allowed to bind in the gut area, and they responsible for integrity of the epithelial cell lines in the gut flora. I mean gut, that is anatomical factors. And another is structural functions: wound healing, inflammation control, and the immune system development. For these also, the microflora that supports. And the metabolic functions like vitamin synthesis, metabolic carcinogen, ferment non-digestible residues, and intra-epithelial cell differentiation, proliferation, transformation, and absorption for all this. So in such a way, it governs gut, brain, lungs, skin, all this governed by the normal microflora. So impact of infectious disease on microflora. So when a person suffering from some diseases like HIV, hepatitis, or any other related illnesses, that is disturbing the microflora definitely. So antibiotic resistance, pathogens, immunosuppressive drugs. So any other modern technologies, or when a person suffering from Clostridium difficile infection, there is Clostridium colitis. All this might contribute for the infectious diseases. So the person may be present with any one of these defects, then that leads to disease, while the imbalance of normal microflora will be resulting, even it may be resulting in multi-organ dysfunction symptoms. So gut flora is very important, as I already told. So different range or number of microorganisms will be present in general. So in the stomach it will be less. Its number will be about 10 to the power of 2 to 10 to the power of 3, and the colon have highest 10 to the power of 12, up to 12 microorganisms will be there. So this is depends upon the environment and its the nature of the inhabited uh, site that determines uh, the number of the microorganisms that it presents. Even in the industrial area, more nutrients are available for the microorganisms, so that the microorganisms will be more in the industrial area. So functions of gut microflora, even the gut microflora is present, but it is playing a vital role in the health of disease. So it defends against pathogens in general. If the microorganisms inhabit in the gut, that is not allowing the microorganisms to bind it to the epithelial cells of intestine or gut. Here, some of the normal microflora organisms also produce antimicrobial-like substances like bacterial. So, the microorganisms which is in there, that might be a pathogen, which is likely to be killed. So, another is alterations of intestinal functions. So, even intestine has many functions like motility, sensory functions, barrier functions. So if the normal microflora is properly inhabited, means there is no problem at all. And another is microbiota on the liver axis. So actually the gut microbiota that governs the liver metabolism also. If there is lack of some microorganisms, that may be result in liver carcinoma and inflammation in the liver. Even there is brain gut axis. Even there is no direct contact, some signalings are there. So signaling pathways are there. So through which it controls. Actually, if some person have imbalanced normal microflora, that results in anxiety or depression, even for children, autisms. Even nowadays, many of the children are suffering from autisms. Even many data are there on autisms. Even anxiety, anger, depression, all depends on the presence of the normal microflora in that gut. So when the person is with the dysbiosis, means the person may get anger. When somebody is shouting at us, or if you are shouting at others, that indicates your normal microflora is imbalanced. So for any of the factors, or by any means, the microflora might be disturbed, so that the anxiety, depression might be resulted. So gut microflora influence the neurons, so it disrupts the brain and resulting in autisms or depression or anxiety, something like that. So substances secreted by the microbes that infiltrate the blood vessels and direct to the brains, and through neurocortex that stimulate vagus nerves. So it is indirectly activate endocrine cells. So that is more indirectly that influence the immune cells and inflammation that can affect the brain. So hurtful effects due to the gut microflora is when the imbalanced microbiota is there in the gut, that leads to functional gastrointestinal disorder and changes in the sensory motor functions, and more intestinal permeability, and stress, anxiety, all will be resulting. So in this regard, how it is overwhelmable means probiotics. If you give the probiotics as a treatment, there may be chance to recover from the effects, and antibiotics or fecal microbiota transfer. Actually, the fecal microbiota transfer, it is like that transplantation, like a kidney transplantation, the fecal microbiota transplantation also there. So it is one of the method. It is not usually new one. Even in the olden days onwards, Chinese people, they will swallow the pieces of their own. That is to regain their normal microflora. They did it knowingly or unknowingly, that doesn't matter, but it is possible. Uh, so in the latter slides, we will discuss on fecal microbiota transfer. So gut microbiota dysbiosis leads to chronic liver diseases like cirrhosis and cholangitis and non-alcoholic fatty liver, alcoholic liver disease, and biliary cholangitis. These are some diseases related to the liver when the dysbiosis of the microorganisms that occurs. So microbiome and adaptive homeostasis. So I already told the normal microflora that enhances the immunity. So some of the non-pathogenic microorganisms may mimic as well as the pathogens. So for those, the immunity will be there. Applied or adaptive immunity will be there. Here, the presence of some microorganisms might be normal microflora, that is, or probiotics, that is enhancing our immune cells and immunity. So the naive CD4 cells giving the rise to differentiation into Th1, Th2, or Th17, or T red cells. So a single cell can differentiate into many different cells, so each has its own function. So the CD4 cells, so-called T helper cells, in the presence of segmented filamentous bacteria that secretes RO or gamma T, this induces the differentiation of CD4 cells into Th17 cells. Actually, the CD17 cells is playing role, and they secrete many of the interleukins, cytokines that is essential for controlling the infections. Likely, that Bordetella fragalis, the T-bit, this factor induces the differentiation into T helper cell 1. So this is protection against intracellular microbes like viral infection. Then T helper cell 2, this is protection against parasites uh, and other extracellular pathogens. Likely T cell regulators, which regulates the immune system. So the immune cells and the system is regulated by T red cells. So in such a way, the presence of these, whether fragalis, Clostridium, or segmented filamentous bacteria, these are important for differentiation of these cells into whether Th1 or 2 or 17 or red cells. So if any of these are absent, for example, the fragalis is absent here, so there is lack in protection against intracellular microbes. Likewise, the other defects will be resulting. So the presence of this microflora that enhances the immunity in such a way that can differentiate CD4 cells into other cells of populations. So mucosal and the systemic immune responses. Actually, the mucosal response is the gut response. Actually, it is also important topic. So the normal microflora, even any microbes that present in the environment, that might enter into through some tracts like respiratory tract or through oral gastrointestinal tract. So many of the microorganisms are exposed. So all are will be uh, managed by our mucosal immune responses. So segmented filamentous bacteria that presents that is playing role in the differentiation of the cells and giving the protection in the mucosal responses. Likely, the systemic responses also managed by our microflora organisms. So human gut microbes interactions in diseases. So when dysbiosis results that leads to heart disease, liver cancer, and Parkinson's disease, Alzheimer's disease, that is related with the brain, then prostate cancer, kidney infection, allergy, and obesity, diabetes. These are possible. So, when the disease is resulting, these are the targets in general. On another hand, it is possible to prevent these diseases through prebiotics. It is nutrient for probiotics. So, we have to consume properly the required quantity of prebiotics. And the probiotics is concerned, we have to take adequate amount of the probiotics in the form of certain foods, maybe through the curd, cultured milk, or cheese, something like that, diet, even the food like the bread and others, or fecal microflora transfer. Through this, it is overwhelmable that the dysbiosis, and we can regain the normal microflora. So these will pave the way for treatment and the record from the illnesses caused by dysbiosis. So probiotic protection against Clostridial difficile infection. So actually, how we can prevent through the probiotics? Even when you are visiting physician and consulting with the physician, sometimes they may give heavy dose of antibiotics. They may give along with the lactobacillus tablets. The tablets form, they will give the lactobacillus. So that is to avoid dysentery as well as the disturbing the normal microflora. So likewise, if we give the probiotics for the treatment, it is producing metabolites. The probiotics organisms producing the metabolites like proteases or bacterioses. So Clostridium difficile is endospore former. When they become into vegetative cell in the favorable environment, they produce the toxin. So the toxin is responsible for the Clostridium difficile infection. And generally it is called colitis, Clostridium difficile colitis. So it causes inflammation in the gut or intestine and results in bloody diarrhea, heavy pain in the stomach. That would be resulting. Even this may be also, this type of infection also may be resulting due to the usage of antibiotics. So when the normal microflora is lacked or underwent dysbiosis, this may be predominant. The Clostridium difficile may be predominant. So if we give the probiotics, the metabolites produced by the probiotics like proteases, that will DNHS or kills the vegetative, toxin. DNHS the toxin. Actually, toxin is the protein, so the protein will be denatured by the protein enzyme, while the bacterial or other bacterial like substances that will kill the vegetative cells. So likewise, the probiotics can provide protection against Clostridium difficile infections. So modulation of gut-brain axis by probiotics. So actually, the presence of gut microflora that modulates the brain axis, so that many of the defects like depression or anxiety is overwhelmable, because there are certain neurotransmitters secreted in the presence of the normal flora or probiotics, and the short chain fatty acids, the neurotransmitters. So these are essential for normal functioning of the nervous system. So when there is dysbiosis, there should be difficult. Actually, for example, in this dysbiosis, so it has leaky gut or epithelial cells, so that outpouring of many of that intestinal contents to outside, and that all contributes for the inflammation. So that affects the brain function. While if the balanced normal microflora is there, or probiotic organisms are there, that is, short chain fatty acids will be more, and also the neurotransmitters will be secreted. So these will govern normal functioning of the brain, so that gut-brain axis modulation is possible by probiotic. So probiotic uh, will be resulting, probiotic using will be resulting in normal functioning of the nervous system. And the probiotics on the skin, it's actually even some of the lotions are mixed with the probiotics. So they are uh, mentioning the cream is available with the probiotics. Actually, there is glucose also. So probiotics that protects our skin from other skin infection causing microorganisms. So the presence of the skin lotions or with the probiotics, that is better. That would be giving protection. So fecal microbiota transplantation. So actually, this is the new concept in the treatment. When the person suffering from dysbiosis, or when the person suffering from colitis caused by Clostridium difficile, then there is a need of replacement of normal microflora. In case of colitis patient, definitely there would be imbalanced normal microflora. So it is necessary to replace with the fecal microbiota. Even the fecal microbiota might be differs according to the region, according to the continent or country, anywhere. Because I know uh, in Africa, most of the African people, they will eat raw meat. Raw meat they will eat. The same we can't. Even I read some report, some of that single cell protein uh, based on algae, that is only consumed by the Japanese people. Even if we consume it, we may face to digest. So that is not acceptable by our system. So likewise it differs. So anyhow, the fecal microflora transplantation is going to be practiced in the near future, and also the banks will be available in future, like fecal flora bank or fecal microbiota bank. Here we may pay and collect that consortium of fecal microflora, and we may take as a treatment. Actually, for this donor and donor is must, even as usual like other donors, like blood donors or organ donors. Here the donor should be free of the disease. For example, like HIV, hepatitis, or any other colitis or any other intestinal infections. So the person should be healthy, and the person should be free of certain diseases, so they can be eligible for donor, so he can donate. So from that healthy donor, through the intestinal tubing or coloscopy or gastroscopy or enteroscopy, the fecal matter can be withdrawn, and that could be kept in the uh, bank, in the frozen condition, or any other preservative method can be followed to maintain it. So likewise, it is possible to generate the fecal microflora bank, and those who are needy, they can request and they can get the fecal microflora to record from their players. So potential mechanism of fecal microflora transplantation includes microbiome modulations. So by direct interaction or competition, the microbiome modulation will be resulting on microflora transplantation. So that will confer the health benefits. Then quorum sensing. So affect microbiome behavior and the community composition. So this is also will be enhanced, and host bile acid metabolism alteration will be there, and the host immunity modulation will be there. So all this can be achieved when a person in need of replacement of normal microflora, fecal normal microflora means that is possible. So these are very much essential, that fecal microflora, even the gut microflora, which is very, very important one, because that determines all the activities of a human. So in future it is possible the engineered probiotics as living medicine, it is possible. So the probiotic organisms may be given along with that other microorganisms, and or may be subjected to engineering genetically, and that can be given for the certain treatment. So for all this, it is referred to well known about metabolomics, metaproteomics, and the metatranscriptomics of the probiotic states. So we are in the conclusion part of this topic. So it is very, very important to have the healthy food, even prebiotics, probiotics, and also another term is called symbiotics. Actually, symbiotics is both the combination, prebiotics and the probiotics. Suppose if you consume probiotics alone, there is no prebiotics means that is also difficult. Why? Because there is no proper nutrients for the probiotics. So probiotics also require some nutrients, so that is achieved from the prebiotics. So prebiotics rich food should be consumed, so we can enhance the inhabitation of the probiotics. So in the day-to-day life, we are taking knowingly or unknowingly in the form of probiotics, maybe the bread or curd, buttermilk, like that, even pickles. So through this we are acquiring. So newborn babies, they may acquire through its mother's milk. So likewise, all the stages we are acquiring, so certain modulation will be there. During the childhood, the intestine will be developed, so that would be determining the child health and its growth also. So we need to have all this healthy food, and we need to live healthy life. So whenever it is possible, add the probiotic rich foods, prebiotic rich foods. Even it is generally said that the greens and vegetables we have to consume more, so that can be a prebiotics. So always have the healthy foods and the probiotic rich foods, and we can live the healthy life. So thank you one and all for your patient hearing. So I am very happy today, because since it is the World Health Day, the topic also related to the same. So you might have enjoyed my session, and if you have any queries, please post it. Thank you.
Thank you, sir, for sharing your knowledge on probiotics for future development. Let food be the medicine and not medicine be the food. Let's make time to have a healthy lunch. Participants are requested to join the session like by 1:50 p.m. to start the session. Thank you, sir.
முன்னாடி ஆன்சர் பண்ணிட்டதா ஆன்சர் பண்ணிட்டேன் இல்லையா?
Hello. Hello. Yeah. Actually, in general, the fungus is not present in the gut region. Bacteria only will be present, but it is very, very rare case, and it is commercially available. The Aspergillus-based fungus. I mean, it is fungus. Aspergillus-based probiotics are there, but it is not much very frequently available. Yeah, it is already listed now, like Lactobacillus, Propionibacterium, and Lactococcus, and bifidobacteria. I have listed already, right? Even including Saccharomyces. Saccharomyces is the yeast.
Thank you, sir. Participants are requested to join to the respective sessions linked by 1:50 p.m. to start the presentation without any delay. Thank you.